welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner. Hello, and welcome to our event. We will get started in just a few minutes. Audio for this event is being provided using your computer speakers. Telephone audio is not being provided for this event. Please ensure your speakers are not muted and you have adjusted your volume accordingly. Please use the Q&A pod or the chat pod to ask questions of the presenters. Type your questions at the bottom of the pod. Time permitting, your questions will be answered at the end of the session. If you would like to make the presentation slides full screen, click the outward 4-arrow button in the upper right corner of the pod containing the slides. To return to normal view, click the inward 4-arrow button in the upper right corner.
Good morning and welcome to the last day of the Regulatory Education for Industry Annual Conference 2023. And of course, welcome to the second day of CBER track. This conference is conducted in collaboration with the FDA's Small Business and Industry Assistance Program as an educational event on regulatory topics in medical product development. Today, you will hear more presentations from the Center for Biologics Evaluation and Research. But before we start our presentations, let me first take care of the administrative and continuing education information and disclosures. As was already mentioned, this conference offers over 32 interprofessional continuing education credits. The obtained certificate of attendance may be used in support of credits for the Regulatory Affairs Professional Society, the Society of Clinical Research Associates, Association of Clinical Research Professionals, and Society of Quality Assurance, as well as for continuing education credits for physicians, nurses, and pharmacists. In order to claim your CE credits, all participants must attest to their attendance and complete faculty evaluations and the final activity evaluation via the CE portal, ceportal.fda.gov. For multi-day activities, participants must attest to their attendance and complete the faculty evaluation each day. final evaluations must be completed within 2 weeks after the activity, no exceptions. The last day for final evaluations for this year's event is June 23rd. In addition to the need to demonstrate attendance, please complete your conference evaluations because we put a lot of faith in these evaluations. They guide us in our planning for future educational events, and we greatly appreciate your feedback, comments, and suggestions. Now, each track of the conference has its own survey and evaluation link, and each night SBIA program has been sending, and tonight they will send again an email to all of the attendees who logged in today to listen to our sessions. The email will have information on the CE claim codes and will contain the survey and evaluation link. CE claim codes will be sent only to those attendees who have registered and logged into the live broadcast on the Adobe Connect platform for the day when you are claiming attendance and for each track that you have attended. And now to the disclosures. All faculty are expected to use generic names. If trade names are used, those of several companies should be used rather than only that of a single company. Continuing education faculty speakers are required to disclose when products or procedures being discussed are off-label or unlabeled, meaning they're not FDA approved, and if there are any limitations to the information that's being presented. All CBER speakers who are presenting today have nothing to disclose. And finally, we can start our morning sessions. As you've seen in the agenda, the first 3 sessions this morning will be about product quality and manufacturing for biological products. Oh, here it is on the slide, and you can see it right here. And our first session will be moderated by Dr. Graham Price. So without further ado, Graham, please take it away. Thank you.
Thank you, Larissa. My name is Graham Price. I am a team lead and CMC reviewer in the Office of Gene Therapy in OTP. It's a great pleasure to introduce our first speaker today, Dr. Ramjay Vatsan. Ramjay is an Associate Director for Policy in the Office of Gene Therapy in OTP within CBER FDA. He joined CBER in 2006 and has served as a team leader in the Gene Therapy branch, as an acting branch chief in the Cell Therapy branch, and is currently the Associate Director of Policy. Prior to joining FDA, he worked in basic and translational research at the National Cancer Institute and Washington University in St. Louis. Dr. Vatsan has extensive research experience in viral and bacterial vector development, gene delivery, and cancer immunotherapy. He is an expert in the CMC aspects of cell and gene therapy and has co-authored several regulatory articles and CMC guidance documents. Dr. Vatsan is an ASQ certified quality auditor and full-time CMC master reviewer. So let's get on with the talk today. Please, let's start the first presentation. Thank you.
Good morning. My name is Ramjay Vatsan, and I'm an Associate Director for Policy in the Office of Gene Therapy in Office of Therapeutic Products in CBER. During this talk, I will provide information about the CMC development program and how sponsors of CBER regulated products, specifically cell and gene therapy products and vaccines, can obtain additional guidance to get the chemistry manufacturing controls ready for licensure. In this talk, I will provide a background on the need for CMC development readiness program. We will discuss the eligibility requirements to participate in the program, how to apply to participate, and how the applications will be selected. At the end of this talk, you will have a clear idea of what CDRP is, who is eligible, and how the program can facilitate accelerating CMC development, and what are the next steps in terms of goals of the program beyond directly helping participating sponsors. There has been a huge growth in the interest in cell and gene therapy clinical studies, with 199% increase from 2016 to 2020. Many of these products are rapidly advancing towards later phase clinical studies and towards licensure. With the rapid pace of clinical development and early encouraging clinical study results, sponsors are thinking of ways to advance their CGT products and are taking advantage of clinical programs that provide additional benefits in terms of obtaining FDA's comprehensive advice on clinical studies for products that can treat rare diseases and conditions with unmet medical needs. These comprehensive programs include RMAT, Regenerative Medicine's Advanced Therapies, and Breakthrough Therapies designations. The requirements for RMAT and Breakthrough are slightly different, and I refer to FDA's guidance on these topics for additional information. Due to early evidence of clinical benefit with CGT products, the number of applications to be enrolled under Breakthrough and RMAT programs have steadily grown over the past few years and can be clearly seen from this graph. Red is for RMAT and blue represents Breakthrough Therapies. Given the growth in Breakthrough and RMAT designations and the use of innovative trial designs, master protocols, and phase 2 studies that become licensure studies, and with the consequent expectation in the rapid development of clinical studies, CMC development tends to lag behind, and this has the potential to result in delays in product approvals. Please note that there is no change in the CMC expectations and requirements. In order to assist sponsors accelerate CMC development to keep pace with the clinical development, FDA is initiating a CMC development readiness program. The CDRP program is designed to assist sponsors prioritize CMC development early to prevent delays in product approvals. The program is designed to assist CMC development of product sponsors who are prepared and motivated to get their product to the market. Sponsors with expedited clinical development programs can benefit from additional interactions with the agency. Some of the CMC challenges that the sponsors may face that the CDRP program is expected to address is listed in the slide. This is not an all-inclusive list, but serves as an example of challenges that may be addressed during the pilot. The examples include CMC help related to vector manufacturing, purification, lot release tests, bioinformatics, stability studies, assay validations, and issues related to these topics. The CMC Development Readiness Pilot Program went into effect on the 1st of April 2023, and a Federal Register notice was published as shown in the slide. CDRP aims to encourage and facilitate CMC development, increase communications, and resolve CMC issues, and this is expected to remove CMC-related issues with product approvals and provide earlier patient access to life-saving therapies. A total of 9 INDs will be selected each year under the CDRP program, with 6 INDs from CBER and 3 INDs from CDR. These will be selected each year. Just wanted to emphasize that it will be selected each year, and 6 from CBER and 3 from CDR. A link to the FR notice is included in the slide. The main goals of CDRP are encourage sponsors to take initiative for product development earlier in their clinical development program and not wait till clinical development is complete. FDA will provide 2 dedicated CMC Type B meetings to provide product-specific guidance. This will be in addition to other multidisciplinary meetings that FDA offers to sponsors of breakthrough and RMAT designated products. There can be additional follow-up discussions to address any lingering questions after the Type B meetings. This schematic provides an overview of the CDRP process flow. Starting with the request to participate, applications will be selected and the sponsors notified. Sponsors will have to submit meeting packages for the Type B meetings, and there will be opportunities for follow-up discussions. These CMC-focused meetings will be in addition to other PDUFA meetings such as end of phase 2 or pre-BLA meetings. In the next few slides, we will go over the eligibility criteria and the selection process. Minimal requirements to participate in the CDRP program are listed in the slide. IND is an active commercial IND. The IND has been submitted in or converted to eCTD format unless the IND is of a type granted waiver from eCTD format. IND should be before the end of phase 2, but there can be exceptions to this rule. I will discuss those exceptions in a later slide. There are certain exceptions to the requirement that the IND be in a clinical development phase prior to end of phase 2. The sponsor should have a commitment to pursue CMC development plan that aligns with expedited clinical development program. Combination products that require extensive inter-center, cross-center communications are less likely to be selected in the pilot. There are certain specific differences in the eligibility requirements between CBER and CDR. For example, CBER files, the requirements include breakthrough and RMAT designations in addition to the files being related to either cell and gene therapy products regulated by Office of Therapeutic Products or vaccines that are regulated by the Office of Vaccines in CBER. CDR specific eligibility criteria, the minimum eligibility requirement is that the product should have an IND with CDR and meet the breakthrough or fast track requirements, though the files may not yet have obtained that designation. FDA will make a final eligibility determination on a case-by-case basis. As I had explained earlier, exceptions to the prior to end of phase 2 rule would provide certain files that are already in phase 3 access to the CDRP program. This again will be determined on a case-by-case basis. FDA may grant files at a later phase clinical study. For example, files that are under the CID program. CID program is a complex innovative trial design program designed for clinical studies that are at late stage product development and are not for first-in-human clinical trials. I recommend that you refer to FDA website for additional information on the CID program. The request to participate information should include at a minimum the following information. The request to participate to the appropriate center should be submitted as an amendment to the IND. Provide some background information such as breakthrough RMAT designations that have been granted or any other active INDs that you may have. If you have other approved products, please describe these products in your request to participate and provide reference numbers. Include information on any approved products in foreign markets. Describe your level of manufacturing experience, similar products or different product types that you manufacture. If you plan to use a CMO, a contract manufacturing organization, please provide a brief description of their manufacturing experience. Describe your CMC development plan. This we will go into detail in a later slide. Is the IND on hold for CMC reasons? If so, please describe the status of the hold. Some of the files that may be on hold for CMC reasons may still be eligible to participate in the CDRP program. The proposed plan for your request to participate should include a proposed plan and timing of meetings with FDA, as well as any other meetings and discussions foreseen. What is a CMC development plan? This section of the application should provide a description of the current status of the CMC development. All product-related information such as CQAs that have been identified, drug substance, drug product manufacturing strategy, with a plan for future commercial manufacturing. The plan should be fully thought out and should include plans for future supply chain management, plans for process validation, and how you propose to get your manufacturing process ready for commercialization of the product. We understand that some of these things may be in early stages of development, but we want to make sure that you have a visualized your process for getting a product to the commercial stage, and you're taking appropriate steps to get the product ready for commercialization. So with that, we'll take a quick polling question. Who can participate in the CDRP program? We went over the requirements to participate and what are the eligibility criteria for participating in the CDRP program. So this question is based on the subjects that we have discussed so far. Who can apply to participate in CDRP program? The choices are sponsors who have an active IND, sponsors who have an IND in an eCTD format, or sponsors who have an IND for a product with accelerated clinical development timelines. Give us a few seconds for you to go over these different options. I understand it's a little tricky question. The answer is all of the above. Sponsors who should have an active IND, sponsors should have an IND in an eCTD format, and sponsors should have an IND for a product with accelerated clinical development timelines. So we will go over the CDRP selection criteria. How will the application selected to participate in CDRP program? First of all, there is no deadline for submitting an application. You can submit an application whenever you're ready. The review will occur on a rolling basis, and the sponsors will be informed of FDA's decision to select within 180 days of the application. That doesn't mean we will wait for 180 days to notify you, but that's the maximum time that it might take. FDA intends to consider the following things: anticipated clinical benefit of facilitating earlier patient access to the product, novelty of the product, complexity of the product or its manufacturing process. Please note that sponsors with experience with a particular product type or class or type of manufacturing process would be less likely to be selected. If you have a lot of experience with manufacturing, you may not need additional coaching to get your product to the clinic, to the market. Less experienced sponsors, less experience with manufacturing a particular type of manufacturing process may be given additional consideration while selecting applicants. Sponsors will be required to sign a disclosure agreement. The disclosure agreement is to ensure that a public sharing of knowledge gained during the CDRP program to help other sponsors who are at earlier stages of product development. Shared information may include case studies. It will be discussed and agreed upon between the sponsor and the FDA. The information that will be shared will be discussed and agreed upon as part of this disclosure agreement between the sponsor and FDA. A disclosure agreement letter template will be sent to the sponsors prior to final selection in the CDRP. There may be examples of type of information that will be discussed and disclosed. The disclosure agreement will be designed to share generalized information to help initiate discussions during the workshop. And we just want to confirm that no disclosure of confidential information will be made without agreement from the sponsor. CDRP related information is available on the FDA website. Sponsors can also reach out to FDA's CBER point of contact for additional information or clarifications. If a question is related to CDRP application that was submitted, then the best point of contact will be the RPM assigned to that IND. Once the IND is accepted into the CDRP program, one should submit a meeting request as an amendment to the IND. The meeting request should include a reference to the CDRP program, a set of specific CMC questions, a meeting background package that provides background information pertaining to the questions, and it is highly recommended that a complete meeting package be submitted at the time of meeting request. To summarize the CDRP process flow, to expedite review process after that application is accepted, we recommend submitting a Type B meeting package with a meeting request. This process should be followed for both Type B meetings and for follow-up discussions. And the approximate timeline for these meetings are shown in the schematic. The Type B meeting will follow FDA's regular Type B meeting timelines. That is, it will be scheduled within 60 days of the request. We highly recommend submitting the meeting package with the meeting request. Sponsors can request meeting format, written or teleconference. FDA may change the meeting type depending on the nature of the topics to be discussed. However, expect a teleconference to be the standard format for Type B meetings under CDRP. When a teleconference is granted, it will follow the standard Type B meeting format. FDA will send out a written response to the sponsors' questions typically 2 days prior to the meeting. A meeting minute will be generated by FDA and issued within 30 days of the meeting. Follow-up discussions. Follow-up discussions are for questions arising out of Type B meetings. The follow-up discussions may be in the form of teleconference or written response. Sponsors should send a written request with questions as an amendment to the IND for follow-up discussions. The cover letter should mention CDRP in the subject line. Background related to the questions should be included with the request. If it's a direct follow-up from FDA responses or clarification questions, sponsors should reference the FDA communication. Follow-up discussions will be scheduled for Type D meeting timelines within 50 days. Please note, the follow-up discussions are for questions arising out of Type B meetings, and follow-up discussions may be denied if deemed inappropriate by FDA. So to summarize the topics discussed so far, and to provide an opportunity for you to think about the topics discussed, here is a question. What benefits can CDRP participant expect to get from the program? options, unlimited number of meetings with FDA review teams, advice on clinical trial design, 2 CMC-focused meetings with FDA review team with a limited number of follow-up discussions. So the answer is number 3, 2 CMC-focused meetings with FDA. These are type B meetings with the FDA review team and a limited number of follow-up discussions. So at the end of this pilot program, towards the later part of the pilot program, we expect to organize a workshop and follow it up with a strategy document. The goals of the workshop will be to promote innovation and understanding in the CMC development readiness of products under expedited clinical development. Lessons learned through the pilot may be presented by FDA in a public workshop as case studies. Sponsors of INDs who participated in the CDRP program will also be asked to present their CDRP experience and lessons learned. Lessons learned may also include those from CMC development of products that have not yet been approved by FDA. FDA intends to follow up with strategy document focused on lessons from the CDRP and based on the proceedings of the workshop. So in summary, the CMC development readiness pilot is a FDA initiative to assist in CMC readiness of products under expedited clinical development. CDRP's aim is to help sponsors meet product safety and consistency standards for novel products designed to mitigate or treat serious and life-threatening diseases or conditions. It's aimed at increasing communication between FDA and sponsors and to remove any bottlenecks in the CMC development program. CDRP started on April 1st, 2023, and will continue till the end of PDUFA-VII period or 2027. A total of 9 applications will be accepted per year during the pilot. 6 of them will be for CBER regulated products and 3 for CDR regulated products. Sponsors of INDs with accelerated clinical development timelines are invited to apply to the CDRP program. The goal of CDRP is to provide patients with earlier access to new drugs and biologics. So with that, here's our contact information for additional studies, information, and you can reach out to us, or you can go to the FDA website to obtain more information. Thank you.
Thank you for that great presentation. So we have a few questions that have come in. So let's start with this first question here. So slide 13 provides CBER-specific eligibility criteria. How do CBER eligibility and selection criteria differ from the criteria for CBER? Over to you, Jay.
Good morning again. Thanks for that question. So there is a slight difference between CBER and CDR eligibility criteria. The intent of both the programs are the same. That is, we want to make sure that products that are under accelerated clinical development are given additional support to make sure that they reach a stage where they are not inhibited because of lack of CMC information in the biological license application or new drug applications. So we want to make sure that the sponsors have additional opportunity to interact with the agency reviewers. So that's the intent. Now, the requirements for CBER is slightly different in the sense you are required to have a prior designation of breakthrough and RMAT designations. Breakthrough or RMAT designations. The breakthrough designations and RMAT designations are required before you submit the CDRP request because it provides us clear indication that your product is at an accelerated development pace, and it has already been reviewed by the clinical reviewers and has assessed the need for additional help. Now, in CDER, this is taken on a case-by-case basis. They will be looking at, again, they will be reviewing the clinical information at the time a CDRP request is submitted, and they will assess at that point. So the requirements are still the same, but slightly different. I hope I made that clear. Thanks. Over to you, Graham.
Thank you. So the next question we have is, part of the information requested for CDRP applications is to provide approximate dates for when the 2 additional Type B meetings are needed. But it's challenging to provide the timing given development timelines often change and evolve. Can we comment on this? Over to you, Jay.
Thanks. We do understand that this is going to be a difficult thing to predict all these things. So the products are under development, and we expect that there will be a lot of challenges in getting the product, understanding what the product needs are and what are the questions. The questions may not even be there fully. So we do understand that there will be a difficulty in deciding what timings are needed and when the questions would be asked. But what we ask is that you provide an approximate time when you would like to have a meeting, so that it'll help us plan and get our resources ready for interacting with you. So this is just approximate time. So at the time of requesting, so you would have an opportunity to make changes if that's what is needed. Thanks.
Okay, thank you for that answer. So the next question we have is the completion of the disclosure agreement required for both CDER and CBER applications? Jay.
Yes. So yes, the disclosure agreement is required for both CBER and CDER. It's the same disclosure agreement. The purpose of the disclosure agreement is to make sure we don't inadvertently disclose information that you may not want disclosed. So the intent of this program is to educate others who participate, who may not be participating directly in the program, but who would be watching us, who would be interested in learning from our experience, your experience and our experience. So the intent of the program is to provide additional support and education to sponsors who are participating, and as well as those who are not participating directly, but who might be under the same accelerated timeline or who are expected to be in the same accelerated timeline. So we do have a similar requirements for disclosure agreement, and the disclosure agreement is to make sure that we, whatever we disclose is agreed upon between the sponsors and the agency. Thanks. Over to you, Graham.
Thank you. So how many cell and gene therapy files will be selected during the CDRP program? Jay.
That's a good question. So we, now, according to the CDRP program, we expect to accept about 9 applications per year. So 9 applications between CDER and CBER. So out of these 9 applications, 3 will be for CDER and 6 will be for CBER. And out of these 6 CBER applications, we expect 5 to be applicable to CBER on cell and gene therapy products and 1 for vaccines. But of course, these numbers can be slightly modified if there is an additional need from CBER as compared to CDER, or if there is additional need on the CDER side, we could adjust the numbers. But the total number that will be accepted per year will remain constant. That will be 9 applications per year. Thanks.
Okay, thank you. So what is the deadline for submitting applications to participate in the CDRP? Jay.
So there is no real deadline. So the intent of this program is to make sure that whenever you're ready, that you would send us application and request to participate in the program. So with that motivational thing, so we would expect you to submit any time that you're ready, and we don't have a particular deadline to accept the applications. It'll be reviewed on a rolling basis. As and when you submit an application, we'll start reviewing it. Over, Graham.
Thank you. So the next one is, can I submit an application for breakthrough or RMAT designation at the same time as CDRP? Over to you, Jay.
No, we want it to be the CDRP program can only accept in CBER, only accept applications that have a prior designation of breakthrough or RMAT. So we would recommend that you submit the breakthrough or RMAT application first and get that cleared, and then submit a CDRP application. Thanks.
Okay, thank you. So the next one is, if I am not selected during a year, will my application be considered during the next review cycle? Over to you, Jay.
So that's a good question. The intent of this program is to make sure that everybody is accepted into this program, but we have a limited number of space. So if your program is not accepted, you will get a declined letter. And once you get the declined letter, you'll have to resubmit your application for the next round. Thank you. Graham.
Okay, so the next one, oh, that's actually a good question. When is the CDRP workshop planned to be held, Jay?
That's a good question, of course. So the intent of the workshop is to educate everybody else and get a program where the sponsors get an opportunity to discuss their experience with the program and share their experience with others. And also FDA can share its experience with others. So with that goal in mind, we expect this workshop to be somewhere in 2025 and towards the end of 2025. And after the workshop, we will have a strategy document drawn. The strategy document will be to summarize the results of the workshop and to decide how this would proceed into the next PDUFA program. Thank you.
Okay, so the next question is, if the application is declined, will the declined letter include feedback to the applicant regarding the reasons the application was declined?
Yeah, that's we intend to provide a certain amount of feedback, but of course, one can reach back to the agency and ask if you have additional clarification questions, because this is done as part of the IND. So you would be able to talk to your product office-specific and product-specific project managers, and they would pass on the questions to the reviewers who may be able to answer your questions. So we do expect to send a limited amount of information if you are declined. But as I had said earlier, the intent of this thing is to encourage sponsors to get their product into the market as early as possible. And we will do our best to make sure that all applications are accepted as appropriate. Thank you.
Thank you, Jay. The next question we have is, what are the chances of a program that does not have breakthrough or fast track to get approval for CDRP? Over to you.
If you do not have a breakthrough or RMAT and you have a cell and gene therapy or a vaccines product, I think the best way to do it is you should apply for one of these designations first. If you have a drug is your thing, which is regulated by CDER, then you can submit both of these things together, and then that will be reviewed in CDER. But if it's a CBER regulated product, like a cell and gene therapy or a vaccine, the best process would be to get your breakthrough or RMAT designation first. Over to you, Graham.
Thank you, Ramjay. And I think we have time for one last question, which is that in one of your slides, you state that sponsors should include any approved product information in the application. Can you comment on the rationale for inclusion of this information?
So that's a good question. So one of the reasons for asking for this information is to get an idea. Sometimes these applications, these marketing authorizations may be done outside the country. If it's in the US, then that shouldn't be a problem. But if you've gotten a marketing authorization in some other country, we would like to know about it. And also that would give us some idea of your expertise and experience with manufacturing, commercial manufacture of such a product. Thank you. Over to you, Graham.
Okay. Thank you very much. I think that concludes the question and answer session for this presentation. We're going to go to break a little bit early, but we will restart at 9:25 a.m. for the next presentation. So that's in around about 15 minutes. Okay, so we'll reconvene then. Thank you all. Welcome back. For those of you just joining, my name is Graham Price. I'm a team lead and CMC reviewer in CBER's Office of Gene Therapy, and I will be moderating this session. It's a great pleasure to introduce Dr. Aaron Seeto. Aaron's a CMC reviewer of cell therapies in the Office of Therapeutic Products in FDA's Center for Biologics Evaluation and Research. Dr. Seeto obtained his PhD in chemical engineering at Auburn University with a research focus in cell biomaterial interaction. Prior to joining FDA, he worked as a research scientist at the Marcus Center for Cell Characterization and Manufacturing in Georgia Institute of Technology, researching therapeutic cell characterization for predictive clinical application and improvement of therapeutic cell manufacturing. In 2018, he joined FDA's Office of Tissues and Advanced Therapies, which is now the Office of Therapeutic Products, and has reviewed a wide variety of tissue-engineered products, cell-based therapy products, including stem, and somatic cells, extracellular vesicles, and lyophilized tissue products. Let's take it away with your presentation, Aaron.
Good morning. I would like to first thank SBIA for this opportunity to speak today. My name is Aaron Seeto. I am a product reviewer in the Office of Therapeutic Products in the Center of Biologics Evaluation and Research, or CBER. Tissue-engineered products with a cellular component are complex, and there are lots of challenges during their product development. In this presentation, I'm going to talk about some CMC considerations for tissue-engineered product development from a CBER perspective. Here's my presenter disclosure. I have no conflict of interest to disclose. This presentation reflects my view and it does not bind or obligate FDA. My goal today is that by the end of the presentation, you will be able to understand the advantages and CMC challenges associated with tissue-engineered products throughout product development. Understand the advantages and challenges associated with expedited product development programs. And to be aware of key FDA guidance documents and other CMC resources available for manufacturers to assist in the development of tissue-engineered products. Before I start, I would like to give a brief introduction to my office, the Office of Therapeutic Products, or OTP. OTP is one of the product offices within CBER in the FDA. The mission of OTP is to protect and enhance public health through the regulation of biological and related products, including gene therapies, gene-modified products, tumor vaccines, stem cells, human tissues for transplantation, plasma products, bioengineered tissues, and medical devices with biologic output. Products that are regulated under OTP is very diverse. This slide expands a little bit more on the different subcategories of the types of products that I mentioned in the previous slide. For this presentation, I will focus on tissue-engineered products. In the next two slides, I'm going to give two examples of tissue-engineered products that are regulated by OTP. The first example is Macy, manufactured by Vericell. Macy was approved under a Biologics License Application, or BLA, in 2016. It is a cellular sheet that consists of autologous cultured chondrocytes on a resorbable porcine collagen membrane for implantation into cartilage defects of the knee. In this product, there is a cellular component, which is the autologous cultured chondrocytes, and also a scaffold component, which is the porcine collagen membrane that the chondrocytes are seated on. The final product is supplied as a 3 cm by 5 cm sheet, and the surgeon will trim the product to the size and shape needed to cover the damaged area. More information can be found on the FDA website. Another example is StrataGraft, that is manufactured by Stratotech. StrataGraft was approved under a BLA in 2021. It is an allogeneic cellularized scaffold product that contains cultured keratinocytes and dermal fibroblasts in murine collagen. It is indicated for treatment of deep partial thickness burns. Stratigraph is supplied as a 100 centimeter square rectangular sheet and may be trimmed to fit the shape and size of the wound area. Again, more information can be found on the FDA website. With that, I am going to go over some general background on regulatory review of tissue-engineered products in OTP. During regulatory review, there are several applicable regulatory frameworks for tissue-engineered products. Most tissue-engineered products contain a human cellular component, as you see in the previous examples. So they must follow the regulations that are outlined under 21 CFR 1271 for human cells, tissues, and cellular and tissue-based products, or HCTPs. They must also follow the biological product regulations under 21 CFR 600, as well as the drug regulations under 21 CFR 312 for investigational new drug and 21 CFR Part 210 and 211 for CGMPs. Scaffold that are used in tissue-engineered products are considered as device constituent. So device regulations under 21 CFR 800 will also apply, specifically including quality system regulations under Part 820. And because oftentimes the biologics component is combined with the device component, that is a scaffold, as the final product. So that would be considered as biologic device combination products as a single entity, and therefore regulations on combination products under 21 CFR Part 3 and 4 would also apply. The regulatory decision-making process in the pre-market and post-market review at the FDA are based on a benefit-risk assessment, and every regulatory decision involves a unique risk-benefit assessment for the disease, patient population, and agents being evaluated. This assessment is informed by science, medicine, policy, regulations, relevant scientific literature, and judgment. Essentially, it's a data-driven decision-making process. Regulatory review at FDA is highly product-dependent, and this is particularly true for products regulated under OTP. Manufacturing scale is one of the biggest reasons. Some products can be manufactured to treat thousands of patients per lot, for example, in allogeneic products. But some product can only be made to treat just one patient per lot, like we see in autologous products. Even with a similar manufacturing scale, the manufacturing procedures, technologies, and methods of different products can differ widely. Comprehensive testing is challenging for products with little test material or very short shelf lives. Therefore, risk of product depends greatly on source materials and how the product is made. Also, high inherent variability of some product types makes demonstrating manufacturing comparability and consistency very challenging. As you may know, there are multiple stages in the product development life cycle. The stage of product development guides the review concerns, and of course with safety being the primary concern at all stages. It goes without saying that safety and quality must be designed into the product. Although potency and assay validation are not required until later stages, we highly encourage sponsors to qualify or validate their assays and processes and develop a potency assay early in the product development. A typical early product development may look something like the diagram that is shown here. There will be a significant effort in the initial product characterization where pilot manufacturing and preclinical testing are conducted to help understand the critical quality attributes, CQAs, and critical process parameters, CPPs, that are specific to the product. These information are used to establish initial product specifications and manufacturing process. Depending on the results of the preclinical studies, adjustments to the specifications are likely needed. All these early product development and preclinical data are going to be used to justify safety and quality of the product for use in clinical studies. We acknowledge that product safety and quality may not be sufficiently understood at this stage. Further refinement of specifications is expected during clinical trials, and FDA encourages continued improvements to product quality, but the improvements or changes must be balanced with maintaining product consistency. For the FDA to assess the safety and quality of the product, the sponsors need to assure that product lots used in preclinical testing is representative of the actual clinical lots. By the time the sponsor is ready to move the product forward to clinical trials, the sponsor should have adequate information on CMC, pharmacology, toxicology, and clinical information to be included in an IND submission. In terms of CMC, here are some information that the sponsors should provide. A description of composition, manufacture, and control of the proposed IND product. Sponsors should also describe the testing being conducted to assure identity, quality, purity, and potency, if applicable, of the product. There should be also batch data to demonstrate the sponsor's capability to consistently and reproducibly manufacture the product. Also, there should be information on product stability, storage, shelf life, container closure, labeling, and tracking information. For INDs that are going into late-stage clinical development, we expect that sponsors have a controlled manufacturing process. That means there is a sufficient knowledge of the manufacturing process to determine critical process parameters and be able to set in-process quality criteria for action limits and rejection limits. This would also be the time to have a plan for future commercial production, whether it's through scale-up or scale-out. We also expect that sponsors have well-developed and qualified or validated analytical assays. In particular, a biologically relevant potency assay should be in place prior to initiating a phase 3 study or clinical studies that are intended to support a marketing application. We also expect that sponsors have sufficient manufacturing experience from the early phase clinical trials to refine their product acceptance criteria. And that would be a brief overview and expectation on the regulatory review throughout the product development lifecycle. In the next few slides, I will go over some manufacturing considerations for tissue-engineered products. In general, tissue-engineered products consist of 2 or more regulated constituents and produced as a single entity. So they involve unique considerations to ensure that each respective product constituent is safe and function as intended. This is very challenging because product developers not only have to identify appropriate testing for individual constituent, but also for final products after assembly of all the components. This slide summarizes a high-level manufacturing considerations on product testing. Most of the time, the cellular component is obtained from a cell source and then undergoes cell processing and manufacturing steps prior to combining with the scaffold. If allogeneic cells are used, the sponsor must perform donor eligibility screening and testing to reduce the risk of transmitting relevant communicable disease agents and diseases. If cell bank system is used, there should be information regarding the master cell bank history, source, derivation, and characterization, including cell bank testing to adequately establish the safety, identity, purity, and stability of the cells. In-process testing should be conducted to evaluate the product at multiple stages of manufacturing. Testing and characterization of cellular component should include a measure of safety, identity, purity, and potency. In terms of scaffold, there is a wide variety of matrix materials available. For example, biologic materials, synthetic materials, or processed tissues that are derived from human or animal sources. The appropriate testing depends on the scaffold, but in general, information to establish safety of the scaffold may include sterility, endotoxin, and biocompatibility. Depending on the scaffold characteristics and intended use, there are FDA guidance documents available providing clarifications on what biocompatibility testing are needed. Because the design and properties of scaffold has impact on the characteristics of the final product and manufacturing consistency, it is important to have sufficient testing to determine the physical and mechanical characteristics of the scaffold. Again, product developers have to determine the appropriate testing depending on the intended clinical use of the scaffold or final product. But some examples of physical characteristics for testing may be mass, volume, density, and porosity. For mechanical characteristics, that may be tensile strength, elastic modulus, and fatigue, etc. Appropriate testing for performance may be needed as design control during product development. For example, many tissue-engineered products are designed to degrade or resorb after implantation to allow tissue regeneration to occur in vivo. So the resorption profile of the scaffold should be properly characterized. After combining the cells with the scaffold, you should consider any in-process testing necessary to evaluate any impact on the cell-scaffold assembly due to cell-scaffold interactions. And before the release of the final product for administration, release testing should be conducted on the final formulated product. Important attributes to measure might be similar to those for the individual components. For example, sterility, mycoplasma, endotoxin, product dimensions, identity, purity, viability of the cell's population in the scaffold, and product maturation and degradation, etc. When conducting testing or characterization on tissue-engineered products, the sampling method may utilize one of the approaches listed here. The first one is testing on surrogate samples, where sample product is made using identical materials and manufacturing method, ideally manufactured at the same time as the clinical product. For example, a smaller dimension of the product that is manufactured at the same time. In this case, additional data may be required to demonstrate that the surrogates are representative of the final clinical product. The second approach is using a portion of the clinical product. Sometimes there are extra or unused portion of the clinical product available for testing prior to administration, for example, the edge of the product. In this case, the sponsor must demonstrate that the portion of the product is representative of the whole clinical product. The third approach is separating the cells from scaffold so that the cells and scaffold can be evaluated individually. For example, the tissue product is dissociated with enzyme to recover the cells. In this case, the impact of the dissociation process should be carefully considered. And the last approach would be using the entire product for lot release testing, which we generally see for testing in sterility, potency, endotoxin, mycoplasma, identity, etc. Lot release testing should be conducted on final product after all manufacturing steps. For example, if the product undergoes additional culture period for the product to mature after combining the cells with the scaffold, then lot release testing should be conducted on the product at the end of the culture. There are many manufacturing challenges for tissue-engineered products that should be considered during the product development. This slide listed the ones that are very common to biological products with cells. For example, limited product manufacturing experience prior to licensure, which leads to incomplete knowledge of critical process parameters. Critical quality attributes not entirely understood due to limited characterization of drug product, drug substance, and in-process material. Assays not fully developed and qualified. Product variability arising from source materials. Reproducibility of replacement cell banks. Increased demand for qualified reagents and materials. Limited product testing due to limited material available or short shelf life. Limited stability data. Complication in planning for advanced manufacturing, process automation, and scale up or scale out. There are also lots of challenges when conducting comparability studies due to the lack of reliable reference standards and validated assays. Oftentimes, there is also a great challenge when autologous product fail to meet release specification and it has direct impact on patient. Here are some manufacturing challenges that are unique to tissue-engineered products. And one of them is the development of appropriate in vitro and in vivo testing and characterization methods. And this is due to a unique set of characteristics in tissue-engineered products. For example, complexity in 3D structure, heterogeneity in composition, small manufacturing lot size, expected remodeling of the product post-implantation. So the final product specifications determined through in vitro testing may not be predictive of clinical safety and efficacy because most tissue-engineered products are generally designed to undergo in vivo remodeling after administration, meaning that the product is not meant to stay the same in the body. And defining potency of performance requirements is also very challenging as tissue-engineered products may have multiple modes of actions, and product performance is specific to both product type and intended use. For example, product indicated for cartilage repair may require testing to assess the capability of forming cartilage tissue or supporting a certain compressive load, whereas vascular grafts may require biomechanical testing to assess the ability of the product to tolerate repeated access without leaking and or withstanding a certain burst pressure. All these challenges may also hinder the comparability assessment when there's manufacturing changes. Speaking of comparability assessments, we understand that manufacturing changes will be made throughout the product development. Manufacturing changes are common in late-phase studies. Some manufacturing changes include scale-up, tech transfer, change in manufacturing platform or scheme, reagents, starting material, formulation, etc. When significant manufacturing changes are made, comparability studies may be necessary to demonstrate that the pre- and post-change product are comparable. However, it is to note that comparable products are not necessarily identical products, but instead, the quality attributes of the product is highly similar before and after change, so that there are no adverse impact upon quality, safety, or efficacy of the product. Manufacturing changes can have impact on many aspects of the manufacturing process. Here are just a few considerations for product developers to think about. Let's say you have a change to the cell source. You may have to look all the way back into donor eligibility, cell bank qualifications, cell bank capacity, etc. One point to consider is that manufacturing changes can be implemented at any stage of the product development, but the potential impact of a manufacturing change on the extent of comparability testing can increase the farther you're along in the product life cycle. Here are some GMP considerations regarding product development. For products that are in phase 1 clinical trials, there's more flexibility in how GMP compliance is achieved, and you may find more details in the guidance as shown in the bottom of this slide. Since the manufacture of tissue-engineered products is complex, we acknowledge that the suitability of a facility depends on the nature of the product, and not all state-of-the-art facilities are ideal for every product. It is to note that manufacturing under GMP does not automatically mean that you will have the best quality product. GMP may improve the product, but mostly adhering to GMP will allow you to control product quality and safety and to help ensure manufacturing consistency. Also, GMP cannot prevent manufacturing errors from happening, but can help ensure that controls are in place to catch them and take appropriate corrective actions. Another aspect to consider is to align clinical and product development. For example, after an IND is allowed to proceed, some may think the clinical trials will take months or years to complete, and there will be more than enough time to make necessary adjustments to manufacturing. However, this is probably not the best way to look at this, since product development is usually very time-consuming. And instead, product developers should always think about the readiness of their product development for situations where there are promising clinical results observed during the early phase study, and they want to accelerate the clinical development and advance to late-stage clinical trials. Here are a few considerations for product developers. It is not advisable to begin studies intended to support licensure if they are undecided about what manufacturing process will be. We recommend that product developers do not underestimate the time and resources needed to bring manufacturing up to level of phase 3 and commercial production, because some product development are particularly challenging and time-consuming, like establishment of quality attributes, measurement of potency, and demonstration of product stability. One thing to always keep in mind is that to approve a BLA, all assays and methods have to be validated, and the facility has to be ready for commercial production, and this takes lots of time and resources. In the next couple slides, I'm going to talk about the expedited product development programs that FDA has developed to support product development. To expedite development of promising treatments, FDA has developed multiple expedited programs over the years, including accelerated approval, priority review, fast-track designation, breakthrough therapy designation, and regenerative medicine advanced therapy or RMAT designation. You may be more familiar with the first 4 programs since they have been around for quite some time. The RMAT designation program was developed to implement certain provisions of the 21st Century Cure Act that was signed into law in 2016. In order to be eligible for the RMAT designation, the product has to meet the definition of regenerative medicine therapy, which is defined as cell therapies, therapeutic tissue-engineered products, human cell and tissue products, or any combination product using such therapies or products. Combination product can be eligible for RMAT designation when the biological component provides the greatest contribution to the overall intended effects of the combination product. And it also applies to certain gene therapies, including genetically modified cells that lead to a sustained effect on cells or tissue. To be eligible for the expedited programs, there are different criteria that have to be met for each program, but one common criterion is that the products are indicated to treat serious condition. And to receive accelerated approval, the product should appear to provide meaningful advantage over available therapies and demonstrate an effect on either a surrogate endpoint or an intermediate clinical endpoint. To receive priority review, the product would need to demonstrate its potential to be a significant improvement in safety and/or effectiveness. For fast-track designation, Non-clinical or clinical data would be needed to demonstrate the potential to address unmet medical need. For breakthrough designation, preliminary clinical evidence would be needed to indicate that the product may demonstrate substantial improvement over available therapy on one or more clinical significant endpoints. And lastly, for RMAT designation, preliminary clinical evidence would be needed to indicate that the RMAT product has the potential to address unmet medical needs for such disease or condition. Here is a table summarizing the different features or benefits of these expedited programs. With accelerated approval, the application approval is based on surrogate or intermediate clinical endpoints, so it saves time in the approval process and reduces waiting period for patients to obtain clinically meaningful benefit. With priority review, the review clock is shortened to 6 months as compared to 10 months under a traditional review clock. For a product with fast-track designation, sponsors can have more frequent interactions with the FDA. It is also eligible for priority review and rolling review. For breakthrough products, sponsors can have all the benefits of fast-track designation, as well as more guidance on the product development, and FDA will commit more resources from senior management. And lastly, for RMAT products, sponsors have all the benefits of breakthrough designation, as well as early interactions to discuss any potential surrogate or intermediate endpoints and potential ways to support accelerated approval. Here are some CMC expectations on tissue-engineered products with expedited development. In general, clinical program advances rapidly for breakthrough and RMAT products. Timelines from early to late development may be compressed. However, it should be kept in mind that accelerated clinical development does not change CMC and CGMP regulatory requirements and expectations. Product development needs to focus on all CMC and CGMP's issues early. For example, CQA, CPPs, assay and process development and validation, raw material qualification, and supply chain establishment, major manufacturing changes, etc. Planning for commercial scale manufacturing, including comparability studies, should be conducted as early in phase 1 and phase 2. So aligning CMC with clinical development is crucial. The goal of these expedited programs is to ensure the availability of a quality product that can be consistently produced at the time of approval. FDA may exercise some flexibility on the type and extent of manufacturing information that is expected at the time of submission or approval for certain components to a certain degree on a case-by-case basis. And it depends on product characteristics, seriousness of condition and unmet medical need, manufacturing processes, robustness of quality system, and strength of the risk-based quality assessment. Areas of potential flexibility may include validation strategies, manufacturing scale-up or scale-out strategies, use of post-marketing commitment or post-marketing requirements. Besides the expedited product development programs, there are also other interaction opportunities with our office before submitting an IND. For example, we have pre-IND meeting, which provides a non-binding, formal scientific discussion between all CBER OTP review disciplines and the sponsor. The goal of a pre-IND meeting is to achieve a successful IND submission. The sponsor will submit questions to us on areas that they want our feedback, and we will review the relevant information and respond to the questions. We typically grant one pre-IND meeting for each product. We also have interact meetings, that is previously known as pre-IND meeting. The goal of interact meeting is to provide informal consultation for innovative investigational product at an early stage of development on issues that are not yet at the pre-IND meeting phase. For your reference, here are some very useful links to FDA guidance documents and other CMC resources that we frequently refer to our stakeholders. To summarize some of the key points on manufacturing considerations for tissue-engineered product, tissue-engineered products are complex and require assessment of the cells, scaffold, and the final cell scaffold product. Depending on the tissue-engineered product, final product release testing may require use of part or entire final product. When only parts of the final product or surrogate product are used, sufficient supporting data may be necessary to demonstrate that they adequately represent the clinical product. Finally, while there are lots of challenges during the development of tissue-engineered products, FDA has developed expedited programs and interactive opportunities to support product development. We recommend that product developers seek FDA advice early and throughout product development. And here are some contact information for your reference. Thank you for your attention, and I'm happy to take any questions.
So thank you for that wonderful presentation, Aaron. So questions are starting to come in from the audience, but I'd encourage anyone else with any questions to please type them into the chat pod and take the time to state the question clearly. So the first question that I think that has come up is this one. Does CBER OTP regulate all tissue-engineered products? And if not, how do sponsors determine which center or office to send their submissions to? Aaron.
Hi, Graham. Thank you for the question. First of all, tissue-engineered product can include a wide variety of products, and classification of this type of product can get very interesting in some cases. So the reason I say this is because the classification of a product ultimately determines which center has the primary jurisdiction of the product. So the classification is usually determined based on the primary mode of action of the product. For example, if the primary mode of action of a device biological combination product is attributed to the biological product, then CBER would have the primary jurisdiction of this combination product, and then the regulatory pathway is likely going to be IND, BLA pathway. So essentially, the primary mode of action of a product determines which center the submission goes. And I would like to add that FDA has a product jurisdiction program. The Office of Combination Products assigns review responsibility for combination products to a lead center, CBER, CDRH, or even CDER. So if you are unsure about the classification of your product, I recommend sending a request for designation, RFD, or pre-RFD to the Office of Combination Products, and they will be able to help you to determine which center you should send your submission to. And details are available on the FDA website, including how to prepare for an RFD or pre-RFD submission. If you do a web search on FDA request for designation, then you should be able to find the website. Thank you.
Thank you for that very clear answer. So the next question we have is, for allogeneic human tissue engineered products, is viral inactivation required if donor screening is in place? Aaron.
Yes, thank you for that question. So I think that depends, and it really depends on how the product is manufactured. So if the starting material used are from human sources, then they should have complied 21 CFR 1271, to make donor eligibility determination. So in that case, I would say there's no viral inactivation is not needed. But let's say if animal-derived materials, scaffolds or matrix are used, then I think viral inactivation is likely needed. So that information should be provided in the IND submission. I hope I answered that question. Thank you.
I think so. Thank you so much. So the next question we have is, if one of the biologic product's modes of action is anti-inflammation, is it necessary to identify all the corresponding critical quality attributes? And if all of the critical quality attributes cannot be identified, could an anti-inflammation potency assay be used as the lot release criterion? Aaron.
Thank you for that question. I think that's a tough question and very challenging one. So I think we do accept that, we acknowledge that, you know, the mode of action is very complex for tissue engineered products, and if you think you know, it's the product is is working through anti-inflammatory action, then, you know, assays that specifically look into that may be one of them. But however, I would still say it's really, without knowing more about details of the product and you know, how it works, I don't feel like I can provide a very meaningful comment right now. But I do reference the next speaker, Dr. Matt Klinker. He's going to present on potency assay development. So I think that will be a more useful resource to address this question.
Thank you, Aaron. So the next question we have is there any recommendation from the FDA on the selection of scaffold for the development of tissue engineered products? Aaron.
Yes, thank you for that question. That's an excellent question. So FDA does not recommend what scaffold to be used in the development of tissue engineered products. The sponsor chooses the appropriate scaffold for the manufacturing of their product. And the sponsor would have to submit information on the safety and quality of the scaffold that they choose to the FDA, and we will review the information. So if the scaffold has a device master file, or it is a FDA cleared or approved regulatory files, then the sponsor can provide a letter of authorization from the scaffold manufacturer to allow the FDA to review the information within the master file or the relevant regulatory submission. And a lot of time we would also ask the sponsor to provide a risk assessment for any devices between the cleared, approved, intended use or indication for use and the proposed use of the scaffold in the proposed clinical study. We also strongly encourage sponsor to obtain a written agreement with the manufacturer, so that the sponsor will be notified of any changes to the scaffold, if there's any. And this will definitely help to ensure consistent manufacturing of the tissue engineered products throughout the study. Thank you.
Thank you, Aaron. So the next question we have is, can you speak to any additional release testing requirements that would be expected for materials from animal sources? Aaron.
Yes, thank you for that great question. Sorry, can you hear me? I'm sorry, hold on.
Yes, we can hear you.
Oh, thank you. Yeah, so regarding release testing, so for materials from animal sources. So yeah, so that will be if it's from bovine animal source, then we will look into testing that is for specifically for bovine sources. So let's say if there's I believe it's 21 CFR, 9 CFR. So there are viral testing for bovines. So you will have to provide those testings. And we do have also testing for porcine requirements. So that can be referred to. So I hope that answered that question.
Right, thank you. So the next question we have, I'm not sure we're going to have time for all of this, but can you discuss a little bit about what the common issues and challenges are in establishing release specifications?
Yes, that's a good one as well. So we acknowledge that it's difficult to decide. Sometimes it's difficult to decide what to measure for release testing for the specification, especially for tissue engineered combination products. So some of the common ones would be not capturing key product attributes or CQAs because of, again, complex mode of action and a lot of time lack of complete knowledge of the product. And I would say another common issue is what is the proposed criteria is inconsistent with manufacturing and clinical data. So what we often see is that the criteria set are a very wide range. So this not only add variability to clinical study, but also it may be more difficult to qualify assays and processes. And this can contribute to misinterpretation or over-interpretation of data because of that variability. And I would say there are usually two general considerations right there. So the first one is to determine what needs to be controlled. And the second one is focus on what you want and what you don't want. So for example, if the cellular component is heterogeneous, then you may want to control the cell population in the product and focus on what population you want, and also the population that you don't want in the product. So I hope that answered your question.
Thank you, Aaron. So the next question we have, which may be the last one, is what should sponsors do if tissue engineered product is to be administered fresh, so not cryopreserved, and sterility release testing results are not available prior to administration?
Yes, thank you for that question. We do understand that tissue engineered product can have very short shelf life. So there are circumstances where sterility release testing result is not available prior to administration. So if that's the case, we typically ask sponsor to do essentially three things. The first one is to do an in-process sterility testing on a sample taken 48 to 72 hours prior to the harvest, and with a no growth result from this in-process sterility testing. The second one would be a rapid microbial testing, such as gram stain, on the final formulated product, again with a negative result. And finally, the third thing would be to do a sterility testing on the final formulated product. Since this test result for the final formulated product will be available after the administration. So an appropriate action plan will be required to be in place in case this sterility test result came back positive. So here are some just high-level components that should be included in the action plan, like notifying patients and physicians, identifying the microorganism and testing the antibiotic sensitivity of the microorganism, a description of additional patient monitoring, determining the source of the contamination, corrective actions taken, and also notifying RB and FDA. So that will be it. Thank you.
Thank you. So I'd like to thank Dr. Seeto for that excellent presentation. We're going to go to break now and return at 10:25 when we'll be continuing with a presentation from Dr. Matthew Klinker from Cell Therapy Branch 2 on identifying and controlling attributes related to potency for cell and gene therapy products. We'll see everyone back at 10:25. Thank you. Welcome back. For those of you just joining us, my name is Graham Price. I am a team lead and CMC reviewer in CBER's Office of Gene Therapy, and I will once again be moderating this session. So for our next presentation, it's a great pleasure for me to introduce Dr. Matthew Klinker. Dr. Klinker is a cell therapy product reviewer in the Office of Therapeutic Products within CBER's Center for Biologics Evaluation and Research. Dr. Klinker joined the FDA in 2014 as a postdoctoral fellow in CBER's Division of Cellular and Gene Therapy, where his research focused on developing potency assays and new approaches for cell-based product characterization. He became a product reviewer in 2018 and has since reviewed regulatory submissions for a diverse array of cell and gene therapy products throughout the product development lifecycle. Prior to joining FDA, he completed graduate studies at the University of Michigan, receiving a PhD in immunology in 2013 for his work on cellular immunomodulatory mechanisms in autoimmune disorders. Dr. Klinker has represented FDA at liaison meetings, public workshops, and panel discussions, and routinely presents to industry stakeholders on topics related to development of cell and gene therapy products, including potency assay development and phase-specific manufacturing expectations. His talk today is titled Identifying and Controlling Attributes Related to Potency for Cell and Gene Therapy Products, and I'll hand it over to Matt to get started.
Good morning, and thanks for the introduction. As the slide says, my name is Matt. I am a product reviewer for cell therapy products in the newly reorganized Office of Therapeutic Products. And the topic of my talk today are attributes that are related to potency for cell and gene therapy products and how these attributes can be identified and controlled, and specifically in the context of both developing potency tests for lot release for CGT products, as well as in the context of assessing comparability of products in the context of a manufacturing change. And so the learning objectives, what I hope you'll get out of this talk is, first, I hope you'll understand what we mean when we use the terms potency and comparability in relation to cell and gene therapy products, or CGT products for short. I hope that you'll understand why potency is such a problematic topic for these products, and the specific challenges these products pose to controlling their potency. And then also, I hope you'll be able to summarize FDA expectations for potency assay development at each phase of clinical development. And then finally, I hope you'll have an understanding of what we mean by product characterization and why we recommend product characterization throughout development, and why it's an important aspect of CMC development that can help you meet some of these challenges in later stages of clinical development that crop up around developing potency tests for your product, and as well as assessing comparability. And so that's what I hope you'll get out of this talk. And as a government bureaucrat, I like to start most of my discussions, most of my talks, by citing regulations, of course. And in the sense of not boring you to death, I won't go into too many of the details here. I just want to give you a really a bullet list understanding of what the regulations require as far as potency of biologics, which would include CGT products. And so the word potency in the regulations is defined as the specific ability of the product to affect a given result. And that's a very vague definition. And we generally interpret that to mean the capacity of the product to affect a clinical result or a therapeutic result. So it's the ability of the product to have some kind of clinically meaningful activity in the recipient. And the other two regulations here just simply say that a potency test should be specifically designed for each product, and that for a licensed product, that each lot of a licensed product should not be released until a test for potency has been completed on it. And as I said, I just want to give you a bullet point understanding of it. So the 3 bullet points are that the potency test should, 1, test an attribute related to the product's ability to mediate a clinical effect. It should be designed specifically for the product. And finally, the test should be conducted on every lot prior to release. And so those are really the only regulatory expectations. And as you can see, there's a lot of flexibility there for a potency test. As long as you can meet these 3 requirements, there's flexibility for us there. And although there is flexibility, there are still quite a few challenges that the CGT products face as far as identifying potency attributes and controlling them. And the first really is, as I mentioned, just identifying attributes that are related to potency. It can be very challenging for these products for a variety of reasons. These are very complex products. Cells can do a lot more things than small molecules or proteins or monoclonal antibody products can do. They have many activities. Just the complexity is an order of magnitude larger than you see for other classes of pharmaceuticals, and that really adds to the problems here that many of these activities may be relevant, but the vast majority of the activities a cell product may have are likely not relevant to any kind of therapeutic effect it has, and so it can be challenging to really characterize the mechanism of action and determine which parts of what activities the cell has are actually relevant. And then also cells, there's intrinsically just a lot of variability, both in the starting material. If you're using cellular material from different subjects, from different donors, you have donor to donor variability. And then at the end of the manufacturing process, as I mentioned, cells are just very complex. There's just a lot of variability, and that can be a challenge too when you're trying to identify which attributes of the product are actually important for any therapeutic or clinical effect that you see. Secondly, an issue with products that are composed of cells is that a lot of times a loss of potency might not be immediately reflected in, say, a physical attribute of the product. So if your product is just a protein, that binds to a target antigen, you can measure, and if that binding activity is gone, the product is likely not potent anymore. Whereas with if you're measuring viability in a cellular product, the product may have received an insult that really did hurt the potency of the cells there, but that might not manifest itself in apoptosis or loss of viability as measured by, say, a dye exclusion method. And so sometimes a loss of that quality can't be determined immediately based on just the physical attributes, and sometimes it only would manifest itself later. Additionally, many CGT products have very short shelf lives, in some cases on the order of a few hours, and this very short shelf life really limits the amount of time you have available to test those products for lot release. And so if you have an assay that requires several hours of incubation or several days even, that can be just something that's not feasible or realistic for some products. And so the types of assays that you can actually perform on these products for lot release is limited in some cases by these very short shelf lives. And then finally, these manufacturing processes that are used for CGT products are very complex. They can be very expensive. They can use a lot of materials, a lot of consumables, and in a lot of cases, the entire product lot is intended to be used for treatment or for use in clinical studies. And in this case, some of the material that may be available for testing may be limited just because of these considerations for these products. And then finally, CGT products really are at the cutting edge of pharmaceutical development, and we don't have the benefit of well-established reference standards and controls for many of our products. And in that case, that can limit the types of assays you can use for testing potency, in the sense that you can't compare to, say, a well-characterized, well-established standard, but instead you're comparing it to previous lots that you've made, so you're doing a relative comparison. And so that also provides a challenge, or presents a challenge in measuring the potency of these products. And just to look at at least one of these challenges in a more visual light, I guess. On the left here, we have what's kind of the conventional manufacturing paradigm for drugs and biologics, where you have one product lot, you can make a lot of the product, and the final lot can be used to treat many patients. And while we do see some CGT products that do have this more traditional scale, I'd say much more often we see these more modest manufacturing scales, where one product lot is maybe intended for a few, maybe 10 or 20 patients. And then we also see a lot of manufacturing processes where each individual product lot is only intended for one recipient, for example, an autologous product. And so these different scales, in this more traditional scale, if you use enough product, if you take enough product that is, if you use product that say could treat 10 or 20 patients, and you use that for testing of your product for lot release, that may not have a real impact on the bottom line for you. Whereas for something like these, if you require material that if you used enough material to test or to treat 10 patients for testing here, you're taking a very significant chunk of your product lot for testing, and in some cases, something like that's not even feasible for these. And so this presents another challenge for these types of products. So, I've explained some of these challenges in FDA. We recognize these challenges, and obviously people in industry recognize these challenges. And so in 2011, we published a guidance document. It's called Potency Test for Cellular and Gene Therapy Products. As I mentioned, it was issued in 2011, so over 10 years ago. But this guidance document spells out how we see the problem of potency assays and provides hopefully some understanding of our expectations, and also some advice that we have for product developers and how they can go about developing potency tests. And so I'm just going to go over some of the highlights of this guidance in the next few slides. Again, maybe give a bullet point understanding of the advice here and the information included in this guidance. And I'm going to start with just really the two broad categories of potency tests that are discussed in the guidance. And the first broad category are called biological assays or bioassays. And these assays are assays in which the product is being tested within a living biological system. So you're measuring the product's activity in relation to a biological system, and that can be, in some cases, it can be animal models of disease, it can be in vitro organ models, or even tissue or cell culture systems. And so, just really the key aspect here is that you're measuring an activity of some kind of living tissue, tissue or cells, as part of the assay. And the second broad category of assays are what are called non-biological or analytical assays. And these are this is a much broader category, and these really refer to any test where you're measuring, say, like a physical attribute of your product. So some examples are immunochemical, biochemical, molecular testing. If you're measuring just surface markers for flow cytometry, would be considered an analytical assay. And these don't really measure, say, an activity of it, but they're measuring a more physical attribute of the product. And in cases where these types of assays are used for potency tests, they should be measuring an attribute that has some plausible link to a relevant product-specific activity. So an activity of the product that's relevant to how you think the product works in recipients. And so these are the broad categories of methods. And in some cases, there might be one single potency assay might not be sufficient to really capture or control the potency of a product. And just to go through some reasons why that might be, so why a single potency assay might not be enough. In some cases, there may be multiple active ingredients, or as I've already mentioned, cells have many activities, and some of them may not be related to another, but they both might be, or they all might be relevant to the mechanism of action. And so in these cases, say, or even, if you have two active ingredients, say if you have if there are maybe two cell populations that are both important, measuring the activity of both of those populations may be necessary. The second reason here is the mechanism of action for a cellular product. It may be complex, or it may not be fully characterized. And in both of these circumstances, it may be justifiable to have a broader, or to test more attributes for potency than you would if you had a very well characterized and very simple mechanism of action. Thirdly, many biological assays or bioassays have extensive variability, and it can be hard to develop assays like these that are sufficiently quantitative, that are robust, and have good precision. And in cases where the performance of a bioassay is simply not up to par, or is not as reliable as you would like, or not as quantitative as you would like, this again may justify a requirement for additional potency assays beyond just that potency assay. And then again, as I already mentioned, limited product stability. Some shelf lives are very short, And so a bioassay that requires hours or days to read out is simply not feasible for lot release. And in these cases, it might be necessary to do kind of a broader panel of potency tests that measure other attributes that you can feasibly test on those timelines once you've shown that those attributes you're measuring are actually relevant to the product, to the clinical effect of the product. And so in cases where one assay is not sufficient, we recommend that you use an assay matrix, which really just means a collection of potency assays, whether it be 2 or even more than that if necessary. And so if you're using an assay matrix, in these cases, we generally recommend that if you have a qualitative assay, in general, we don't recommend qualitative assays at all, but if they're unavoidable and you can't improve an assay to make it actually quantitative, in those cases that you really should have at least one or more assays that are more analytical, that have the precision to be considered quantitative, that those are included in your assay matrix. But in general, an assay matrix, it can have bioassays, it can have analytical assays, or it can have both, which really depends on the product. And then really one of the take-homes for understanding a potency assay matrix is that if you're measuring something using an analytical method, so not a bioassay, you really should have data to show that you're measuring something that's relevant, that surrogate attribute you're measuring is actually relevant or has a relationship to a biological activity that actually is meaningful for the product. And so the guidance has these recommendations, but also has a little other recommendations for how you should go about developing potency assays. And as I mentioned previously, the regulations there have quite a bit of flexibility. There's no specific test required by regulations for potency, as long as it's meaningful to that particular product. Really, the flexibility is there for us to really accept anything as long as you're measuring something meaningful. Secondly, the regulations don't require you to actually fully define the mechanism of action, and they don't require that your potency assay mimic a mechanism of action. So that being said, I think it should be evident to everybody that having an understanding of how your product works, of having what you know about the mechanism of action, should be used to inform your selection of attributes that you measure for potency. And so it's useful, even though it's not required, it is useful for potency assay development to have an understanding of your mechanism of action. And then the third recommendation here is that we recommend that you start assay development as early as possible, and that you don't put all of your eggs in one basket, so to speak, by deciding very early in product development what attribute is going to be your potency, be measured and controlled for your potency testing, but instead that you evaluate multiple attributes through product characterization, and you do this throughout development. That you, just for another cliche, that you cast a wide net for potency attributes. And then finally, the assay development, the real bottleneck in development when it comes as far as FDA expectations is that before you start what can be called a pivotal study or a phase 3, the language we use now is something like before the initiation of clinical studies intended to provide evidence of effectiveness to support a marketing application, but really what we mean is a traditional phase 3 study, or something that you're going to use as to cite as the primary evidence of efficacy for your marketing application. Before you start those studies, the expectation is that you have at least one quantitative potency assay in place before you start those studies. And just to elaborate on that, this slide gives an example of the timeline of product development that I think many of us are familiar with, whether starting from preclinical and early stages of development through phase 1, through phase 3, and then up to the licensing application. That potency assay expectations, the two main areas where sponsors get into trouble here is in this area I just mentioned, this transition between phase 2 and phase 3. And we expect at this stage that you have, as I mentioned, at least one quantitative potency assay. And I think the justification, or why we think this is important, really is when you conduct this phase 3 study, having a potency assay in place that's reliable and quantitative and qualified and just something you can actually rely on. This allows you to get a good idea of what the product lots that you use in this phase 3 study look like. And so if your assay is in place and up to speed, at this stage, you can use data from this phase 3 and your assay and your reliable assay, and that allows you to really inform the specifications of the product that you would propose in your BLA. And so it gives you an idea of what the product looked like in those phase 3 studies. And if those phase 3 studies are successful, if they show that the product it does have a therapeutic effect, that you can now have information that you can rely on for the potency of those product lots that were used in that study. And so you can use that information to help ensure that the commercial product after licensing is also just as effective. And then the second place where this trips up sponsors is again at these later stages, and the arrow here is pointing to phase 3. And very often at the end of phase 3, if sponsors have a successful study or they want to move into a BLA, that they want to make manufacturing changes, whether it's scaling up or opening a shiny new manufacturing facility or any variety. There's a lot of reasons that manufacturing changes might be implemented here right before commercialization, and at this stage here, having a good potency test is really important for comparability. And I'll go into the more details on comparability in the second half of this talk. But this is just the two areas where essentially where potency development can really slow down product development if it's not up to speed at these particular stages. And so one of the ways we think that you can avoid those bottlenecks is through product characterization. And broadly speaking, product characterization, when we use that phrase, we just mean that you're looking at many or several attributes of the product to help identify what could be called critical quality attributes. And as defined by ICH Q8, critical quality attribute is really just an attribute of the product that needs to be controlled to ensure a desired product quality. And as it relates to potency, those potency-related CQAs need to be controlled to ensure that the product actually does have a therapeutic effect, but also to make sure that the dosing that you're using throughout at each different stage of development is also are controlled for potency as well. And so what we recommend you doing is, instead of, as I said previously, instead of just choosing very early on what your potency-related CQAs are, and then just sticking with that through development, we recommend that you cast this wide net and test several potential product attributes that may later be shown to be critical quality attributes. And so this diagram here shows the kind of funneling effect that we're recommending here. We're at early stages of development. You measure multiple attributes, and then as you gain clinical manufacturing experience, and then you hopefully can decide which of these attributes are actually the critical quality attributes, and then these are the ones that you will use for lot release for controlling the product later as you get to stage 3, and then maybe even beyond stage 3 into commercial manufacturing. And to just, I guess, describe it a different way, here again is the assay development timeline. And as far as developing potency assays, at the bottom here, we have what it seems to be commonly done by sponsors, where it's a serial assay development. So they start developing assay 1, and for whatever reason they reject it. Either the assay doesn't perform the way they like it to, or the attribute it's measuring, they show that they figure out that's not actually critical to product quality, so they reject it and move on to assay 2, and they reject that one for some reason around phase 2, and then assay 3 and 4 they develop in the aftermath of rejecting those other 2, and the assays 3 and 4 they find actually do measure a critical quality attribute, and they go ahead and validate those assays, and that's what they use for controlling potency. And that's one method. But if your development timeline is shortened, or say you're developing something for a rare disease where where you're not going to treat a lot of patients, and maybe you want to accelerate this this development, or if you try this approach, the serial assay development, you might actually be slowing down. You might not be able to keep pace with clinical development, and in those cases, that would be why we recommend something like this, where you're developing all 4 of those assays at the same time, not necessarily validated or qualified at the beginning, but should be shown to be suitable, at least for what you're trying to do at those early stages. But this allows you to have those assays in place throughout development. And so in this example, you can see assays 3 and 4 were started before phase 1. And so now these assays were used on product lots that were used in those early stage studies. And so you have a much broader data set that you can use to set your specifications and show compelling evidence for comparability if needed later. So you didn't waste time developing these assays later in development. They're already there, and so they're there, and you have a much broader data set with these assays, which will be useful for comparability, as I'll discuss later on. And so just to kind of reiterate why we think product characterization throughout development, why these are important, why this is an important thing to do, and why you shouldn't wait. One thing I haven't mentioned yet here is that at least in early development, it can be hard to know what attributes should be controlled for potency, and a lot of that you might learn in those phase 1 and phase 2 studies. You might learn what attributes are important, or you might learn that you need to have, say, a certain level of potency to actually have a clinical effect. And so, understanding your product attributes before you start doing those very expensive clinical studies gives you a broader basis of knowledge. And instead of putting all your eggs in one basket, you're measuring multiple attributes. And so, if you're only measuring one attribute for potency, you don't really know if it actually is. If it is relevant to the therapeutic effect, or if your specifications or acceptance criteria are stringent enough. In this case, a colleague of mine has compared this to driving a car without a functioning check engine light. So the light isn't turning on, but that doesn't necessarily mean that your engine is fine. And so, if you're going on a very long road trip, just like if you're doing a very expensive clinical study, it would be nice to know that your check engine light is actually linked to some meaningful attribute of your car's engine, right? And so this is just another way to set yourself up for clinical success. And then I've already mentioned assay development, so I won't go into the details too much here, but I will again reiterate that for potency testing, we really expect that you should have at least one quantitative assay for potency in place before you initiate those phase 3 or pivotal studies that are intended to be primary evidence of efficacy for your product in a marketing application. And then again, as I'll go into more details later, I want to talk more about comparability. Having a well-characterized product and having all that information on multiple attributes for your product can really come in handy later when you want to show that the product that you're going to make in, say, your shiny new manufacturing facility for commercial manufacturing. When you want to show that product is comparable to the product you use in your phase 3 and your phase 2, it'll be very helpful to have all that information from the previous from testing the product lots used in those previous clinical studies to support that. And so a well-characterized product can be helpful in that sense as well. And then before moving on to comparability, I do want to just make a clear distinction between product characterization and what we mean by a potency assay matrix. This is something that I think it's spelled out relatively clearly in the guidance, but I don't know if for whatever reason it seems that some people are frequently confused by this. So I'll just to make sure that we make this clear, a potency assay matrix, when we use the term, we are really referring to a series of assays that are used for testing every product lot and that have acceptance criteria in place for lot release. So these are lot release potency tests. And that is in contrast to product characterization assays, which we generally use that phrase to mean tests that are being done in addition to those that are already in place for routine lot release. And these are generally exploratory and in most cases are not subject to acceptance criteria. So these are really just information gathering tests. And so, later stage, these are still useful, but they don't need to be obviously a qualified assay is more reliable, but they don't need to be validated, and they just need to be suitable for use for what you're intending to use them for. And so that's really the main difference. So a potency assay matrix really refers to potency tests in place for lot release that have acceptance criteria, whereas product characterization are really just information gathering assays that you're using to measure attributes that you may find later are important, or you may find later that they're not important, but it's an exploratory exercise for product characterization. And so now that I've covered most of the things that I want to for potency, I come to a challenge question. And so I'll read the options here and see if you give you a few seconds to come up with the answers. And so the question here is, which of the following statements regarding potency tests for CGT products is false? And so which of the following statements is not correct? Choice A is, FDA guidance recommends evaluating multiple potential potency tests during clinical development. Option B is, federal regulations require that potency tests mimic the product's mechanism of action. Option C is, tests for potency should be specifically designed for each product. And option D is, each lot should be tested for potency prior to release. So which of these statements is not correct? Well, hopefully you were able to identify statement B as the one that is not correct. And so I mentioned previously that there is no federal requirement or federal regulatory requirement that a potency test mimic the product's mechanism of action. I mentioned that understanding the mechanism of action is obviously very helpful when you're trying to determine what attributes you should measure as potency tests, but it is not necessary that the assay itself mimic the mechanism of action, only that it's measuring something that's related to the therapeutic effect of the product. Hopefully you got that right. So moving on to the last part of my talk, I'd like to just discuss potency-related attributes of these products as they relate to product comparability. And product comparability is really something that's most important for managing manufacturing changes. And in most cases, if your product is around long enough and if you're lucky enough to get to pivotal studies and lucky enough to get your product to licensing and commercial manufacturing, on that kind of time scale, manufacturing changes are essentially inevitable. They're going to happen. That being said, they need to be properly controlled, and you need to plan for them so that you can manage them properly to avoid delays in your product development, or for a licensed product to avoid delays in commercial manufacturing changes that might result in a shortage. And so in some cases, the manufacturing changes that will happen, if they fundamentally change the product, if the very significant changes to the manufacturing process or the starting material or the reagents used, something very major like that, it may just not be realistic to show that the product after those changes is actually comparable to the pre-change product. And in those cases, we would have to essentially consider it a new product, and that may require a new IND and a new BLA, essentially starting over. And obviously, that's not in anyone's best interest. So I will focus more on comparability assessments that would happen when you have changes that have a moderate or potentially a substantial effect on product safety and efficacy. And in these cases, you may be able to support comparability by conducting what we call, a comparability study or a comparability exercise. And in these cases, what you're really doing is comparing the attributes of the post-change product to the pre-change product. And how thorough that comparison needs to be to show comparability depends on a lot of factors, but probably the most important factor for us is the stage of product development, and that will really determine exactly how thorough that assessment needs to be to establish comparability. And as with potency, with the developing potency tests, there are many challenges for CGT products, or that CGT products present to comparability. The first, again, is that there's limited manufacturing experience. For rare diseases, you might not make that many lots. In some cases, manufacturing might just be limited for several other reasons, or the material that's available from previous manufacturing is just not there at the end of your, when you want to do the comparability assessment, there might not be material from from lots that you previously manufactured, or enough to really do a thorough assessment of that product. It might be limited in process testing. And so in many cases, if you have limited in process testing, you don't really know, you don't have a good idea of what are the process parameters that are critical for your process, and that can be a challenge. So if you make a manufacturing change in the middle of, at an intermediate point in your manufacturing process, you might not have good ideas of what those intermediates already look like because of this limited in process testing. Limited product characterization. If I've already sang the praises of product characterization, but if it's not done, if you don't have a well-characterized product, if you don't know what your CQAs are, it is very challenging to do a convincing comparability assessment if you don't know what's important, what the attributes of your product are that are critical. I don't want to say impossible, but it is very challenging to do that convincingly if you don't have good product knowledge. And then finally, if assay development is limited, if you don't have good, reliable, qualified assays, if your potency assays if you haven't shown that they're actually stability indicating, meaning that they measure something, an attribute that actually goes down when the product no longer works, that can make a comparability assessment challenging. And again, there are almost no well-characterized or well-established reference standards that are widely recognized reference standards for these products that you can compare your pre-change and your post-change product to. So ideally there would be some kind of standard you could purchase from a recognized entity, and you can compare your pre-change and your post-change product to this standard and show that they're both the same as the standard. But the reality is that doesn't happen very often for our products. And so the challenges that I've described the challenges here. So now I'm going to go into, I guess, the details of how you can conduct a comparability assessment. And I think the most important document here that I'm going to be summarizing is an ICH quality guideline. It's Q5E. And I'm just going to start with, what does the word comparable really mean? Like, what do we mean when we say that a product is comparable to another product? And so in Q5E, what they mean, or I guess how it's defined, how comparability is defined here is that the pre-change and the post-change product are quote unquote highly similar, and that this high similarity, that you know enough about the product, that you have enough knowledge about the product that you can determine that those attributes you've shown are highly similar, that is sufficient to ensure that the product safety and efficacy of the post-change product is comparable to that of the pre-change product. So the standard here is not identical. It's not nearly identical. It's highly similar, which of course is vague and open to interpretation. But we hopefully there's some statistical considerations both in this guidance and then that we can give in direct interactions with sponsors to maybe to spell that out in more specific terms. But just to emphasize again, the standard here is that the products are highly similar to be considered comparable. And the guidance or the quality guideline, the Q5E, mentions that if you can show product comparability based solely on quality considerations, and by that it means if you can show just by measuring attributes of the post-change product and the pre-change product, just measuring attributes of those products, if you can show comparability through those analytical studies, that additional evidence from non-clinical and clinical studies may not be necessary. But if you can't do that, then additional evidence, either non-clinical or clinical studies, will be needed to demonstrate that the product is as safe and effective as the pre-change product. As I'm sure I don't need to explain to any developers out there, that is a situation that I think most people would like to avoid. And so I will just focus on these analytical comparability considerations, where you're trying to establish comparability just based on measuring attributes of the pre-change and the post-change product. And for those studies, I'll just go through a couple of considerations that we think are important. Maybe some of the steps that you should take as you're planning these studies. The first step really is if you're thinking about a manufacturing change or proposing one, the first step really should be a risk assessment to figure out what risk that change presents to the product and to the process. And then as part of that assessment, you should look at the attributes that are important to the product quality and the safety and the potency, obviously, and look at those attributes that are most likely to be affected by this change. And based on that, your comparability study should look at those attributes that you know are at risk based on this manufacturing change. And then you need to come up with predefined acceptance criteria for comparability for these attributes. And I won't go into details on that because I think it's outside the scope of this talk, and maybe it would be a talk all by itself. But really, this important part of this step is determining exactly how similar those products that particular attribute needs to be considered comparable. And that can be determined a lot of ways. And hopefully, if you have a lot of manufacturing experience, you can look at historical data, and that can help inform what the distribution of values for that particular attribute look like in your previous manufacturing campaigns, and that can help you there as well. And so this also requires robust statistical methods. It can't just eyeball it, can't just make graphs and look at it and see if they look the same. There needs to be statistics here. And as I mentioned, the thoroughness of this assessment, the thoroughness that you need later in product development is much higher than, say, when you're just getting started. And so for that reason, we generally recommend that if you know you're going to make manufacturing changes before you start commercial manufacturing, we really recommend that you make those changes before you do your pivotal studies. In that case, then you're not making major manufacturing changes after you've shown that the product actually works in that phase 3. And so the bar is just a little lower if you're doing comparability before that pivotal study as opposed to after it, when you don't intend to do any more clinical studies to show efficacy of that product. Then the bar is very high. Other considerations here is that if you can do a side-by-side analysis with sufficient lots to do statistical analyses, that is definitely preferable. And by side-by-side, I just kind of illustrated here, if you have a starting material and you can split this material and you can, in parallel lanes, do the old process and then do the new process. And so you have the product being made at the same time, so you can compare these two in real time, as opposed to, say, an old process which was done at a manufacturing facility that closed 5 years ago, and you can no longer repeat this, and you're pulling product out of a freezer that's been in a freezer for 5 years and comparing it to product that you are now making. So that, there's caveats there. I'm not saying it's impossible, but this is definitely a preferred method for showing comparability. If you can do the old process right alongside the new process, that's definitely preferable. And then again, if changes are introduced late in development, it's going to be a lot harder to show comparability. Not to reiterate this again, but analytical comparability, if you can't do it, it means essentially starting over. You're going to have to do new clinical studies or preclinical studies to show that the product is safe and effective again. And so finally, the final point here is that generally we like to see the comparability study before it's actually been completed. And so we can see those predefined acceptance criteria. So we know that they're actually predefined, and we can look at the methods you use to come up with your equivalence windows. And so we like to see this before, and just so we can get an idea of what attributes you're measuring, and also maybe give you an idea of whether we think that your assessment is going to be thorough enough to convince us. And so in general, before you do a comparability study, it's really best to get good feedback from FDA prior to actually doing the study. And then as far as comparing the attributes, one of the most common methods is equivalence testing here, where you're comparing the mean of the pre-change product to the mean of the post-change product. And here, this is just an example graph where they're plotting. These are plotting the mean of the post-change relative to the pre-change product with the corresponding confidence intervals. And then this is the predefined equivalence window here, where if based on your knowledge of the product and manufacturing experience, here hopefully you can get some idea of what kind of difference is meaningful. and which kind of difference is not meaningful. And so if you're using an assay that has a good precision and your product is not all that different from the pre-change product, you might get a result like this, where the confidence intervals are both within this window here, and so you can consider these products equivalent or comparable on this attribute. If your assay is not very precise, you might get into some trouble here, where your confidence intervals for this measurement are actually one or both of them fall outside of this window, and then you haven't really shown that the post-change product attribute falls within this window. And so this really emphasizes the precision of your assays. It's good to have good, precise assays for this, and to test enough lots where you can get these confidence intervals pretty small. And so that's kind of just like one of the most common ways to go about this. And then in the last few slides, I'll just go through the main reasons for manufacturing changes, or the main reasons for doing comparability assessment. The first I already covered. These are changes in reagents. They may be to improve changes in reagents, changes in the process. Any material change you make to your manufacturing process may require a comparability assessment. I already mentioned that the stage of development really will have an impact on how thorough your comparability assessment should be, but also the significance of the change, as well as the risk that your product may pose to subjects. Those also play a factor in how thorough we want to see how thorough of a comparability assessment we want to see. Another common area or another common scenario in which a comparability assessment is necessary are for facility changes. And so in this case, anytime you're moving manufacturing from one facility to another, it's very often accompanied by other changes to the manufacturing process. And this can be a real challenge too, especially if the old manufacturing facility is no longer online. And so in this case, we'll have to do a comparability assessment. But if you can do this before you start your phase 3 studies, that would be beneficial and reduce the thoroughness that's required for comparability. Another scenario is when you have multiple manufacturing facilities, and so in this example, there's 3 facilities all making the same product. And if you have in this kind of scenario, you would have you'd really want at least some comparability assessment at any stage of development to show or to justify that that the clinical data from product made at each of these 3 facilities can be analyzed and grouped together. And in these scenarios, it's also good to just limit the variability between those facilities as much as possible by using same reagents, same SOPs, same qualifications, same equipment, as many sources of variability that you can limit as possible there. And then the third is a little different. Third scenario is a little different. This refers to changes in analytical testing. So whether it's a change in the assay, whether you're using a different method to test the same thing, or if you're opening a new testing facility that's doing the same tests, but just at a new facility. In both of these circumstances, a comparability assessment would be necessary, and you demonstrate that the testing on both that the assay methods are measuring the same thing, and that the methods themselves are comparable by testing the same material side by side. And then if you have multiple testing sites, you would want to test the same material again at both of those sites to show the analytical comparability. And so with that, that's the end of the comparability section of this talk. And so my challenge question here is, according to Q5E, what is the standard for establishing product comparability? Is it A, the products are identical, B, the products are nearly identical, or C, the products are highly similar? And hopefully I emphasized it enough that you picked out choice C as the answer, that products should be highly similar to be considered comparable. And so with that, I will just summarize my talk. I hopefully gave you an understanding of the challenges that CGT products present to potency assay development, but also an appreciation of the flexibility in the regulations that allow you quite a bit of flexibility in what kind of tests you use, so long as you're measuring something meaningful. And then, as I mentioned several times, that identifying CQAs that are related to the potency of products can be very challenging, and so product characterization early in development can help you do that to avoid those bottlenecks around potency assay development going into a phase 3 study, and then also for assessing comparability later in development. And so I thank you for your time. My contact information is here, as well as information for contacting OTP if you have questions, or some other resources here as well. And with that, I will end my talk, and I'm of course happy to answer questions. You can either contact me or at the end of this session. So I thank you for your time, and enjoy the rest of your day.
So thank you for that excellent presentation, Matt. We have a large number of questions, so I think we should just start leaping into them. So the first question we have is, are biological assays and non-biological assays both required for an IND application, and is a biological assay considered quantitative? Matt.
Great, thanks, Graham. So as far as biological assays and non-biological assays being required for an IND, we don't really have specific, I guess, expectations or requirements for the type of assay that you're doing for an early IND. In some cases, a biological assay is probably more meaningful, but it's not required. It's not a requirement to have both of those kinds of assays. So if you can, if you have a meaningful assay that's not a bioassay, but it's still measuring something relevant, that you can make your justification, and that might be okay. And then as far as biological assays being considered quantitative, in almost all scenarios, I think we prefer to see bioassays as quantitative, and I think you as a developer should. It's to your benefit that they're quantitative as well. So gives you a more sensitive assay that can hopefully detect changes, or I guess a reduction in quality that you might not detect with, say, a qualitative assay. And so, yeah, we really prefer to see quantitative bioassays, except in very limited circumstances that a qualitative assay might be okay. Back to you.
Thank you. So our next question is this one. Sponsors often struggle to set potency acceptance criteria for initiation of clinical studies, given the dearth of data available and due to a fear of rejecting what may be a perfectly acceptable product. Can you discuss how FDA approaches this issue during IND review, and how the decision is made whether to push back on sponsors to get them to tighten early acceptance criteria? Over to you, Matt.
Thanks. Yeah, and so setting acceptance criteria, I didn't really cover that too much in the talk, but it's a really important piece of potency testing, because the idea is not just to measure it, but to control the potency here. So and so at the beginning of development, if you don't have clinical data, so if you're talking about first in human studies, at that point, the acceptance criteria can be based on what you know about your manufacturing capabilities. So hopefully you've done some kind of engineering runs or full-scale GMP runs of your products, so you have an idea of what you're capable of. But then also, it's important if you have preclinical or non-clinical studies that are supporting your first-in-human studies. It's important that the potency of the lots you're going to use or the product you're going to use in the clinical studies is consistent with what was used in those non-clinical studies. And so those are kind of the key, key sources of information for setting acceptance criteria at the beginning of development. And then as far as when we push back on sponsors for tightening their early acceptance criteria, I think as far as if it's safety, if there's a safety concern, then 100% of the time we will push back. And then I think maybe pushback. So another scenario where I might maybe recommend that that specifications or acceptance criteria get tightened is if I know that the manufacturing process, if you show me data in the IND that whatever attribute is 90+% of the cells have a particular if every manufacturing lot you made it has like 90% of the cells expressing a certain surface marker, and your specifications are at like 60%, I'm going to at least recommend that you do it. But if it's not related to safety, in general, the product specifications and acceptance criteria are it's really up to you. But yeah, I think it's to your benefit to have tight specifications as much as is feasible at even at early stages. Back to you, Graham.
Thank you. So the next question we have is, can you provide clarification on FDA's expectations for assay qualification where full validation is not indicated? Over to you, Matt.
So as far as assay qualification, I think our general advice is your assay is a qualified assay is much better than one that's not. And so if an assay is worth doing, it's worth doing well, and it's worth having some knowledge or some confidence in that assay that it's measuring something reliably. And so for qualification of a potency assay, when it gets to a point where we might put you on hold if it's not qualified, and that is when you initiate a pivotal study or a phase 3 study. And so that's when qualification would really be expected. As far as when, I guess the question was about where full validation is not indicated. I'm not totally sure what that is referring to, but the expectation is that your potency assay will be validated as well, essentially every assay you have for your product at the time of applying for a license.
Thank you. So the next question that I think came up was, what is the expectation for a potency assay to support release of clinical drug product early in development? Can you provide examples of what types of method may be acceptable at this early stage?
Sure. So, so release of the drug product early in development, it's hard to give an answer for that. That would apply to all CGT products. And so in some cases, if there's a benefit risk question about if there's significant risk to the patients that are going to, or subjects that will be receiving that product, that there should be some assurance there, or at least to balance that high risk, there should be something in place to assure that they're actually getting a product that could potentially work. And so in those cases, we might want to see we have the expectation that you will have something to control potency at early stages. For other products, if the risk is pretty low, if the risk is minimal, there are products where we don't require potency assays for early clinical development. So it's hard, like I said, it's hard to give an answer that would apply to all products. And that's something we evaluate kind of on a case by case basis, is the risk of the product versus what kind of control for potency we want to see to kind of justify that risk. And then the second part of that question was, did I miss the second part? I forgot what the second part was. Sorry, Graham.
The second part was, can you provide examples of what types of methods may be acceptable at early stages?
Oh yeah. So I don't think I want to give examples just so it's not interpreted as being prescriptive. But there's flexibility there, and it's that's something that would be different for every product.
Okay, thank you. So the next question we have is, in a comparability protocol, should acceptance criteria be pre-specified for assays that are not part of lot release? For example, characterization assays and in-process measurements. Over to you, Matt.
Should they be pre-specified? Yes. I think in general, the general answer is yes, that they should be pre-specified. That's really what makes the comparability assessment convincing, to me at least, is that you're having those pre-specified criteria and then you're meeting them. It's a lot easier to talk yourself into, if you don't have those pre-specified criteria, you can make an argument that, well, it's different, but it's I think it's probably okay. And that's really not what we're looking for. But that doesn't mean that you, at every single attribute, or every single attribute you're looking at, not every attribute you're looking at needs to be part of that assessment, or needs to have a pre-specified criteria. So, but I guess it's in the context of the overall comparability assessment. So if you're doing something very thorough, but you have a very thorough list of attributes that are subject to a pre-specified acceptance criteria, but then you have others that are not, that might be okay. So it really would depend on how thorough the assessment is overall.
Okay, thank you. So next we have, are there major differences between analytical comparability approaches for manufacturing changes and for comparing decentralized manufacturing centers? Matt.
So that is a kind of a difficult question to give a specific answer to. So I think Yeah, I don't know if I would say there are major differences, but there are potentially differences. So if you have multiple manufacturing facilities, you can be using the same process. Hopefully you can be using the same reagents. So there can be there's probably going to be differences, but it's hard to compare that to, say, if you do just a very major manufacturing change. And so, I think there might be differences in exactly how thorough we would want that comparability, but there's nothing in general that we would say is different between those assessments. It would just be based on your risk assessment of what the changes are in the manufacturing process versus your risk assessment of what's different between the manufacturing centers. And that would determine the thoroughness that we'd want to see of the comparability. So yeah, I can't say that there are major specific differences and those kind of comparisons that we'd want to see.
Okay, thank you very much. So the next one is, for the requirement for the potency assay be in place prior to a pivotal clinical trial, is it acceptable for a potency assay to be conducted for drug substance release only? And for example, if the drug product contains an excipient that makes potency assays difficult or impossible on the drug product? Matt.
That is a good question. And so some products that have very complicated manufacturing processes, as I mentioned, there's short shelf lives. There are places where maybe the most important, or like a bioassay, that could be done, I guess, upstream of the drug product. And in that case, it would be maybe on the drug substance as well. And so that's something that we can be flexible about. But I think really if it's not the final product, you're still going to have some kind of test on that final product. Whether it's, maybe it's not a bioassay, say if your excipient is not compatible with a bioassay, that you maybe the major testing is done upstream, but there still should be a measurement of some kind of attribute in that drug product that shows that those last stages of the manufacturing process didn't hurt the product potency. And so I guess the answer would just be there is some flexibility here, but our preference usually is that it's the drug product that's being tested for potency. But I guess one other point would be that a lot of times for cell and gene products, the distinction between drug substance and drug product is it's not as clear as it is for some other types of products. So there's some flexibility there, but we prefer that it would be on the final product.
Thank you. So the next one is, for cell and gene therapy products, can potency assay results be reported in a binary form? For example, positive or negative, yes or no, expressed or not expressed. Over to you, Matt.
Oh, so that to me sounds like it's a qualitative assay, and yeah. So we don't have a strict requirement that I guess we don't automatically reject an assay because it's qualitative, but our strong preference is for quantitative assays. And there's not too many circumstances where a qualitative assay is better than honestly, I can't think of a single circumstance where a qualitative assay would be preferable to a quantitative assay. But there are some things that are very difficult to measure, and in some assays, just are hard to get reliably quantitative. And so in some circumstances, there may be we might be okay with a qualitative potency assay, but it would probably have to be as part of a potency matrix. So, if you have an assay that is, as I said, is very difficult to make quantitative, in that case, if that is a qualitative assay, you would hopefully have, as part of your assay matrix, you would have other assays that are measuring attributes that you can measure more reliably as quantitatively. And then those are, kind of, because I don't want to say covering, but, giving more assurance than just the qualitative assay alone. So in some circumstances, I guess that, those an assay that has like a binary outcome like that is qualitative might be acceptable, but it really, as I said, the preference here is that it's quantitative, and those kind of assays would probably have to be part of a potency matrix with quantitative assays.
Great, thank you. So following on from the potency assay matrix, Do potency assay matrices refer to multiple assays looking at the same CQA or MOA, or at different CQAs and MOAs?
That is a good question. So, so I think for very complicated or very complex products like CGT products, that I think usually there are multiple attributes that are associated with potency, and so I guess we kind of focus on those really big, important potency bioassays where you're measuring the activity of a product. But there's other things that are that are important as well. For at least for if your cell product is if your product is cellular based. Those cells should have, they should be alive. They might need to be, might need to have like a low immunogenicity if you intend for them to, stick around in the recipient for a while. And so in that case, an assay matrix maybe should look at multiple CQAs that are all related to potency. And then in some cases, it might be necessary to have multiple assays that are looking at the same CQA. For example, my answer to the previous question, I was talking about maybe an assay is hard to get reliably quantitative. So maybe there's another assay that can measure something similar, but something I guess a related attribute, but it can do it more quantitatively. So I think, but also I do want to emphasize that we don't want you to have to do multiple redundant assays either. And so the matrix, for a particular product, it might have both of the assays, multiple assays measuring at least CQAs that are kind of related to the same MOA, but hopefully not in a redundant fashion.
Thank you very much. I think that concludes the question and answer session. I'd like to close this morning's proceedings by thanking Drs. Vatsan, Seeto, and Klinker for really excellent presentations. I think these have all been very informative. So I'm going to close out this session. We will reconvene from the lunch break at 12:30 for this afternoon session. Thank you all for attending.
Good afternoon and welcome back from the lunch break. We will now continue the track for the Center for Biologics Evaluation and Research. My name is Larissa Lapteva and I come from the Office of Therapeutic Products in CBER, and I will be moderating the next two sessions. So our next session is a very exciting one because we will be talking about innovative trial designs and statistical methodologies that are important for interpretation of product effectiveness, which is really in the heart of drug development. And for this very vital discussion, we have Dr. John Scott today with us, who will speak about some recent updates on two FDA initiatives related to modernizing clinical trial design: the Estimand framework and the Complex Innovative Trial Design Paired Meeting Program. Dr. Scott is the Director of the Division of Biostatistics in the FDA Center for Biologics Evaluation and Research, where he has also initially served as a statistical reviewer for blood products and for cellular, tissue, and gene therapy products. Prior to joining FDA in 2008, Dr. Scott worked on clinical trials in the psychiatry field at the University of Pittsburgh Medical Center. He has authored and co-authored numerous articles on Bayesian and adaptive clinical trial design and analysis, vaccine and drug safety, data and text mining, and benefit risk assessment. He is the CBER lead for the Complex Innovative Trial Design Program and has been heavily involved in a number of FDA statistical policy and outreach projects, including adaptive design guidance for drugs and biologics and guidance on interacting with the FDA on complex innovative trial design. He also serves as a member of the International Council on Harmonization E9 Expert Working Group on Estimands and sensitivity analysis, and he's also a member of the ICH E20 Expert Working Group on adaptive designs. Dr. Scott holds a PhD in biostatistics from the University of Pittsburgh and a master in mathematics from Washington University in St. Louis. He is a fellow of the American Statistical Association and is a past editor of the journal Pharmaceutical Statistics. So please welcome Dr. Scott, and he will start his presentation in a few moments. Thank you.
Hello, my name is John Scott. I'm from the Division of Biostatistics in FDA's Center for Biologics Evaluation and Research, or CBER. And I'm going to be talking to you today about two statistics-related initiatives to advance and improve clinical trial design: the Estimand framework and FDA's Complex Innovative Trial Design Meeting Program. So in terms of learning objectives, one is to describe the 5 attributes of an Estimand. The second is to discuss the role of intercurrent events in clinical trial planning. Third is to identify key aspects of complex innovative trial designs. And fourth is to explain how to initiate interaction with FDA on a complex innovative trial design proposal through the CID meeting program. So I'm going to start with the Estimand framework. I'm going to give some background and details on how it's implemented. So the real background for the Estimand program comes from the missing data problem in clinical trials. It's long been understood that missing data can cause problems in clinical trials and other medical studies. One issue relates to bias. If observations in the study are unavailable for reasons that might be related to their values, for example, if people who are doing badly tend to drop out of the study, this can lead to biased estimates or erroneous decisions. Even in cases where missing data are missing completely at random, having the missing data reduces the power and precision of the study and its ability to produce useful estimates. Back in 2008, as a PDUFA-VII commitment, FDA launched an effort to address missing data in clinical trials. And a lot came out of that effort, but one of the things that was observed is that there seemed to be confusion in the field between missing data and data that might be better described as meaningless. So to give an example of what I mean by that, suppose we're investigating a pain therapy, and you design a trial with week 12 pain score as the primary endpoint. Imagine a subject drops out of the study at week 8. That subject has a pain score at week 12. We don't know what it is. That's missing data. On the other hand, if a subject dies at week 8 in the study, they do not have a pain score at week 12. It's not that they have one and we don't know it. They're no longer eligible to have a pain score. It's a meaningless concept. So we might describe that more as meaningless data than missing data. So what came out of observations like this is the concept of an intercurrent event, which I'll give the formal definition for it in a few slides. But the basic idea is that something that happens between treatment assignment and measurement of the variable that we care about that fundamentally changes the meaning of that measurement. So there are many different kinds of intercurrent events. These are all hypothetical examples. You can imagine they might have occurred in the same trial. Patient 1 here discontinued treatment sometime during the trial due to experiencing adverse events. But the green dot at the end indicates that we do have their final data score. Whatever the variable we were collecting was, we did collect it for this subject, but they discontinued treatment before that happened. Patient 2 discontinued treatment due to lack of efficacy of the treatment, but we did get their measurement. Patient 3 here has 2 intercurrent events. First, the use of rescue medication, that is, maybe their condition worsened during the course of the trial and it was necessary to put them on additional therapy for clinical reasons. And then as a second intercurrent event, this patient died. Having died before the end of the study, we do not have the data measurement for that person. Patient 4 switched treatments halfway through the study. Maybe they switched to the other investigational arm in the study. And then patient 5 had a change in their background therapy at some point during the study, and then was lost to follow-up. So all of these things are potential intercurrent events that either make it difficult to interpret the endpoint at the end of the study or lead to us not knowing the endpoint at the end of the study. So having intercurrent events can really impact what we think of treatment effects and what treatment effect we might be targeting in the study. So as an example, consider that rescue medication intercurrent event example. During a trial, some patients will require additional medication, others won't. So what this does is it creates different potential choices of the treatment effect of interest in the study. So for example, we might care about the treatment effect of interest regardless of whether additional medication is used. That is, a patient who has rescue medication, their scores are just as meaningful and valuable to us as a patient who doesn't need rescue. We also might say, okay, some of our patients needed rescue, but what if we could create a hypothetical scenario in which none of them had the rescue medication? What would the treatment effect have been in that case? So that's a different kind of treatment effect. Or we might say, what if we only care about the treatment effect in the stratum of the population who would not require rescue medication? That's a different kind of treatment effect. So this brings us to the underlying problem that was discovered as people investigated missing data in clinical trials. It turned out that many trials have had at the time and still have had somewhat vague objectives, things like showing efficacy. An objective of that kind leads to ambiguity in how to handle missing data, and it also raises barriers to communication between regulators and sponsors. So it was felt that what was needed was a more precise statement of the exact treatment effect that was trying to be measured in the study, and that's what's called the Estimand. So the word Estimand itself may be somewhat unfamiliar. I'm going to go through a little bit of terminology to try to clarify a bit. So an Estimand for a study is the thing you want to estimate. It's the thing you care about. So this is a concept such as a treatment effect. In most clinical trials, we're trying to estimate a treatment effect. And the thing we're trying to estimate should flow from the overall objective of the study. The estimator, it's a related but different word, That's the statistic that we use to estimate the estimand. So an estimator is a mathematical procedure such as an arithmetic mean. And an estimate is the specific value of the estimator applied to a specific set of data. So an estimate is going to be a number, like 42 or 18%. So estimand is what we care about. Estimator is the procedure we use to get an estimate of what we care about. And the estimate is the number that we get. Okay, so the idea of an estimand culminated in the publication of the guidance ICH E9R1, R1 meaning revision 1 to ICH E9, Statistical Principles for Clinical Trials. And this revision was an addendum covering estimands and sensitivity analyses in clinical trials. So this guidance introduced the estimand framework, and what it really does is encourages sponsors to be very specific about what treatment effect exactly do we care about in this study and how do we go about estimating it. So more specifically, ICH E9R1 defines the estimand as a precise numerical characterization of the treatment effect at the population level. It works to align the clinical questions, the study objectives, the design, conduct, data collection, and analysis of the study. It makes sure that people explicitly address intercurrent events in their definition of clinical questions and also the treatment effect, and distinguishes intercurrent events from missing data. And E9R1 also clarifies the definitions of sensitivity and supplementary analyses and discusses the roles of each. So where estimands fit in the trial design process, I mentioned earlier that estimands really flow from the overall objective of the study. So there's a clinical question of interest, often a question about whether a drug works in a particular condition. And from that clinical question of interest, we construct the estimands. Having constructed the estimands, we can design the study, conduct the study, and then finally analyze and interpret the data from the study. So the study objectives, again, they flow from the clinical question of interest. The estimands flow from the objectives, but the estimands are also can be seen as a really detailed, specific statement of the objective. Estimands consist of 5 attributes: treatment, population, variable, intercurrent events, and the population level summary. So this is one of the key ideas I'm hoping to convey today, is the 5 attributes of an estimand. And I'll be going over each of these in a little bit more detail. So the first estimand attribute is the treatment attribute. The treatment attribute covers the treatment condition of interest and the alternative treatments to which comparisons will be made. It's usually fairly specific. The treatment attribute will include the dose, regimen, route of administration. It'll include any important background care. For example, we might be comparing investigational therapy plus standard of care versus standard of care alone. The treatment attribute will also include allowed concomitant or rescue medications. Other rescue medications might be considered intercurrent events. The population attribute covers the patients who are targeted by the clinical question. This should be very specific. It'll pretty much always include a diagnosis or potentially multiple diagnoses. Sometimes it will include age or other demographic information. And sometimes it'll include baseline clinical characteristics or prognostic factors. For example, we might be interested in the effect in severe disease or mild disease, or in patients who are positive for a certain biomarker. The variable attribute or endpoint, this covers the variable or endpoint that's going to be collected for each subject to address the clinical question of interest. And then the intercurrent event attribute. This one gets a lot of attention in discussions of estimands, not because it's more important than the other 4, but because it's a more novel idea to focus on. Prior to the Estimand framework, people were already well aware that they had to specify a treatment population and variable. But intercurrent events, so this is the formal definition of an intercurrent event. It's an event occurring after treatment initiation that affects either the interpretation or the existence of the measurements associated with the clinical question of interest. It's necessary to address intercurrent events when describing the clinical question of interest in order to precisely define the treatment effect that is to be estimated. It's really important to understand that intercurrent events are things that can happen in clinical practice as well as in clinical trials. So I think when we think about these in the context of missing data, there's a tendency for people to think, these intercurrent events, they really messed up my data set. But in reality, the intercurrent events are a meaningful part of a patient's journey and shouldn't be dismissed as a statistical nuisance. Potential intercurrent events during a trial design should be identified, and a strategy should be proposed for each of them. So, intercurrent event strategies. What's meant by a strategy is the choice that's made on how to address intercurrent events in order to describe the treatment effect that's targeted. And ICHE 9R1 identified 5 different kinds of intercurrent event strategies: treatment policy, composite, hypothetical, principal stratum, and while on treatment. I'll describe each of them briefly. So the first strategy is the treatment policy strategy. This strategy treats the intercurrent event as if it does not change the interpretation of the variable. Data that we collect for the variable are used regardless of the occurrence of the intercurrent event. It's important to note that this, in general, can't be used for terminal events, such as death or, in some indications, surgery. The fact that the terminal event has occurred may mean that the variable at the end of the study truly is meaningless and can't be collected. The composite strategy identifies the intercurrent event itself as an important clinical event that informs the effectiveness of the treatment. So the occurrence of the intercurrent event then becomes part of the variable. The hypothetical strategy is based on imagining a situation in which an intercurrent event that did occur would not have occurred. We set the variable's value to be the value it would have taken had the intercurrent event not occurred. Note that in general, we don't know that value. There are several important considerations when using a hypothetical strategy. One, it's important to be specific about what the hypothetical situation is in which the intercurrent event would not have occurred. For example, is the intercurrent event rescue medication? Are we imagining a scenario in which there's a shortage of that rescue medication? That might be specific. The hypothetical situation also has to be relevant to clinical practice. If we don't think there is any way in clinical practice there would ever be a shortage of the rescue medication, then such a hypothetical would be of arguable relevance. And then finally, the imputation strategy, by which we guess what the variable's value would have been if the intercurrent event hadn't occurred, has to be sound and reliable. Next is the principal stratum strategy. The principal stratum strategy typically seeks to answer the scientific question of interest for the population of patients for whom the intercurrent event would not, or in some cases would, occur on either treatment arm. That is, no matter which arm they'd been randomized to, they would not experience this intercurrent event. It's important to note that this is not the same as the subset of patients in whom the intercurrent event did not occur, because in general, we only observe a patient on one treatment arm, not on both treatment arms. Typically, the principal stratum approach requires strong assumptions to use. And then fifth, the while on treatment strategy. This strategy addresses the scientific question regarding the treatment effect prior to the occurrence of the intercurrent event. It defines the variable as the measured value up until the intercurrent event occurs. In general, this might mean different duration of treatment for different patients. I'm not going to go through these 5 strategies in great detail, but I am going to give kind of a brief example to convey the idea of each of them. So imagine the intercurrent event is the one that we brought up earlier of a patient receiving rescue medication after their condition worsens in the clinical trial. So the treatment policy strategy would say, okay, well, we'll just consider the use of rescue medication as part of the overall treatment policy. It's part of the treatment attribute, and we don't have to take any special action. We'll just use this patient's variable as we would have otherwise. The composite strategy says we're going to make the use of rescue medication part of the endpoint. That is, we're going to consider it in most cases to be a treatment failure. Whatever arm the patient had been randomized to, it wasn't working well enough to prevent them from needing rescue, so it's a failure. The while on treatment strategy says, okay, we have data up into the point where the patient was given the rescue medication. That data directly informs the effect of the treatment up to that point. So that's the data we'll use. We'll just measure the symptoms up to rescue occurs. The hypothetical strategy says, can we estimate what the effect would have been if rescue medication weren't available? And the principal stratum strategy says we should assess the effect in patients who wouldn't need rescue in either arm. No matter what they had been randomized to, they wouldn't have needed rescue. It's important to note that both of these last two tend to require extensive statistical modeling and somewhat strong assumptions. And in general, the fact that 5 strategies exist don't necessarily mean that all 5 are usable or acceptable in all scenarios. They're just different possible approaches across the whole spectrum of intercurrent events. The population level summary is the 5th Estimand attribute, and this is the attribute that provides a basis for comparing treatment conditions. So some examples of this might be a difference between treatments in mean change over time, might be a ratio of the proportion of patients who respond to treatment, or it might be a hazard ratio. So, bringing all of that together, when we're talking about the Estimand framework, we start with the study objectives and the clinical question of interest. We think about what intercurrent events might occur and might be important to interpreting the variable. So that includes things like treatment discontinuation, use of additional or alternative treatments, terminal events, and so on. We specify a strategy for each intercurrent event that might occur in the study, and note that we don't have to use the same strategy for every event in one study. In general, each event should be considered on its own in terms of picking a strategy. And then from all of that flows the 5 attributes: the treatment, population, the variable or endpoint, other intercurrent events, and the population level summary. So one of the things I mentioned at the beginning is that as we started to think carefully about missing data 15 years ago or so, it became clear that there were some things we thought of as missing data that were really more like meaningless data. But there are still missing data. Missing data still exists, and ICHE 9R1 tries to distinguish between data that aren't meaningful due to an intercurrent event and missing data. And it gives a specific definition for missing data. Missing data are data that would be meaningful for the analysis of a given estimand, but were not collected. And it emphasizes that they should be distinguished from data that do not exist or data that are not considered meaningful because of an intercurrent event. So there are a variety of methods for handling missing data in clinical trials. For example, different kinds of data imputation methods. And these can be considered for missing data, but in general are not appropriate for meaningless data caused by intercurrent events. The existence of missing data will often be the cause of sensitivity analyses. So I also mentioned that ICH E9R1 talks a little bit about sensitivity and supplemental analyses. What it does specifically is it defines sensitivity analyses as different analyses to assess different assumptions that go into estimating a given estimand. That is, if you're looking at a different way to analyze a specific estimand, that's a sensitivity analysis. But if you're looking at different estimands, that's what we would now call a supplemental or supplementary analysis. So in general, you might have a primary estimand in a study and a main or primary estimator for that estimand. That estimator might have various assumptions that you might want to explore alternative methods for. Those would be sensitivity analyses. But if, for example, you want to look at a different strategy for handling an intercurrent event, that would create a different estimand, and that analysis would no longer be considered a sensitivity analysis. It would be a supplemental analysis. Okay, so these are some resources for the Estimand framework. The first one is a link to the guidance I've been talking about, ICH E9R1. The second one is some training material that was published by ICH to accompany E9R1. And in fact, I took some of the figures in this talk I've just given from that training material. And the third is a manuscript that was published by FDA authors giving both clinical and statistical perspectives on the implementation of the Estimand framework. So all of that brings us to challenge question number 1. Which of the following is not an Estimand attribute? A, the variable or endpoint. B, the intercurrent events. C, blinding. Or D, the population level summary. I'll pause for 30 seconds before giving the answer. The answer was blinding. Blinding is not one of the 5 attributes of an estimand. The 5 attributes are treatment, population, variable, intercurrent events, and population level summary. Okay, so that was the first part of the talk. And for the second part, I'm going to be discussing FDA's Complex Innovative Trial Design Meeting Program. So first, some background. It's well known that clinical trials really form the backbone of the evidence that we need for safety and effectiveness in order to approve drugs and biologics. Over the years, the costs and complexity of clinical trials, especially late-phase clinical trials, have really ballooned to enormous proportions. As a result, there's considerable interest in developing innovative approaches to increase the efficiency of trials, things like adaptive clinical trial designs, Bayesian approaches, or ways to incorporate external data into trial design and analysis. Now, there can be some barriers to innovation. For one thing, due to the cost of trials and the risk, sponsors are sometimes reluctant to experiment with novel trial approaches. On the regulatory side, regulators are sometimes resistant to change or to novel approaches because of the risk of bias or type 1 error. In cases where innovative approaches have been accepted by FDA for regulatory use, that information is often non-public. especially for products that have not been approved yet. The need for innovation is especially critical in rare disease and serious and life-threatening diseases. So that brings us to the Complex Innovative Design Meeting Program. And the motivation for this program was that sponsors, in order to succeed with an innovative proposal, they might need more regulatory feedback than usual for a more complex proposal than usual. And they might also need the assurance of having sort of buy-in from all aspects of the review process at FDA. On the other hand, on FDA's side, in order to promote the appropriate use of innovative designs, we really need case studies that we can talk about publicly. So these two things came together to lead to the CID, or Complex Innovative Design Meeting Program, which was formed in collaboration between FDA and industry representatives and was part of our User Fee Act commitments from PDUFA-VI and then PDUFA-VII. So the CID program. What this is a joint effort between FDA's Center for Drugs Evaluation and Research, CDER, and the Center for Biologics Evaluation and Research, CBER. What happens is sponsors can submit innovative design proposals and then have the opportunity to engage with agency staff with 2 additional meetings beyond what's normally granted in a development program. FDA will select up to 2 of these submissions per quarter, and then in the future we use the designs as case studies for outreach and education, and coming to an agreement with the sponsors about the public disclosure of this information is a critical aspect of the program. The additional meetings are led by the biostatistics groups at CDER and CBER, but all relevant disciplines are involved, including always the clinical review discipline and others as appropriate, pharmacology, so on. This meeting program started as a pilot program under PDUFA-VI, and under PDUFA-VII, it is another 5-year meeting program, no longer a pilot. In order to be eligible for the CID meeting program, a sponsor must already have a pre-IND or IND number for the medical product included in the proposal. The proposed CID should be intended to provide substantial evidence of effectiveness to support regulatory approval of the medical product. The trial is not a first-in-human study, and there's sufficient clinical information available to inform the proposed CID. And finally, the sponsor and FDA have to be able to reach agreement on the trial design information that will be publicly disclosed. When the FDA is evaluating proposals for the CID program, we're looking at several different things, including the need for clinical trial simulations to assess design operating characteristics, and this is really one of the key determinants of the complexity of the design from a statistical perspective. The therapeutic need and indication, the appropriateness of the trial design for the program, and the level of innovation of the trial design. This is a link to the website for the CID meeting program. There's a lot of information here. So I'm going to go through 3 of the examples of CID proposals that have been accepted into the program and reviewed. There are more examples than this, but these 3 I think are illustrative of a lot of the key things that we see in the program. So the first example is from Duchenne muscular dystrophy. So for those who aren't familiar, Duchenne muscular dystrophy is a rare, serious, progressive neurological condition. It almost always affects boys with rapidly advancing muscle weakness, typically beginning before the age of 5. It's associated with severe disability, morbidity, and early mortality, in most cases before age 30. And there are a lot of challenges to drug and biologic development in this area. It's a rare disease with a small population from which to study. It's difficult to assess change in functional endpoints over short periods of time. And also, especially in younger children, the disease course can be highly variable. So the design of the study that was proposed in this first case example, this was a randomized, double-blind, placebo-controlled phase 2/3 trial. The primary endpoint was change in dystrophin levels from baseline to a specified time point. There was also an important secondary endpoint, change from baseline in a clinical outcome assessment. The endpoints were analyzed via a Bayesian repeated measures model with multiple interim analyses. Bayesian features have been common to most of the designs that have been proposed to the CID program. CIDs are not required to be Bayesian, but that has been an area that sponsors have been particularly interested in. So some of the features of this first case example. This was a Bayesian adaptive design with the following potential adaptations: stopping the trial for efficacy or safety, potentially modifying the sample size, potentially dropping one of the treatment arms, pooling doses if the data suggested it, or changing the randomization ratio. The sponsor also proposed exploring placebo augmentation using historical controls. So in discussions between the sponsor and FDA, a lot of the focus was on areas that needed clarification in the proposal and also on the details of the clinical trial simulations that were used to show the statistical properties of the study design. The second case example was in pediatric multiple sclerosis. In most cases, multiple sclerosis is diagnosed in adulthood. About 3 to 5% of multiple sclerosis patients are diagnosed before the age of 16. When a child is diagnosed with multiple sclerosis, they may be subject to more frequent relapses and be more likely to experience cognitive symptoms than adults who are diagnosed with MS. So there are some development challenges in this area as well. Again, the patient population is not a large population, and the outcomes can be quite heterogeneous. So a design that was proposed for this indication was a randomized, double-blind, group sequential non-inferiority trial using a Bayesian framework that used what are called meta-analytic predictive priors to leverage information from existing external adult and pediatric studies. And when the sponsor and FDA discussed this study design, those discussions focused on the non-inferiority margin, the scope of the search for external databases, and additional simulations for operating characteristics that were requested by FDA. The third case example is in chronic pain. This was a proposal for a randomized, double-blind, placebo-controlled master protocol to evaluate multiple interventions, so different drugs, across multiple pain conditions, all of them different chronic pain conditions. The primary endpoint for this study was pain relief from baseline. And this study had a Bayesian mixed model repeated measures analysis that included the possibility of borrowing patient information from placebo groups within a given pain condition, or borrowing treatment effect information across pain conditions for a given intervention. And again, the study had multiple interventions across multiple pain conditions. Some of the features of this study, there were possible adaptive design elements, including potentially stopping for futility, modifying the sample size, or adding or removing arms for the study. And for this one, regulatory discussions focused on potential drift over time in placebo response, the exchangeability assumptions across pain conditions, and the missing data that frequently can be encountered in chronic pain trials. So in terms of next steps for the CID meeting program, FDA has several commitments for complex innovative designs under PDUFA-VII, which include continuing the paired meeting program, continuing to develop staff capacity to review complex adaptive, Bayesian, and other novel trial designs. We're holding a public workshop on these topics by March of 2024, so within the next year. And we're committed to issuing a guidance document on the use of Bayesian methodology in clinical trials for drugs and biologics. These are several resources for complex innovative trial designs. The first is the Complex Innovative Trial Design Meeting Program website, which contains a variety of information about the program, including how to apply, where to get more information, case examples, and a variety of other information. Next are direct links to 3 of our case studies that have been written up. And again, a critical part of the CID program is FDA's agreement with sponsors to be able to disclose design elements to serve as sort of teaching examples and case studies. So one of them is in lupus. I didn't discuss that one today. Another one is in lymphoma. And then the third one is the chronic pain example that I did discuss today. We have 2 relevant guidances. One is a guidance on interacting with the FDA on complex innovative trial designs for drugs and biological products. And the other is the guidance on adaptive design clinical trials for drugs and biologics. And then there's a manuscript that was published, I think a little over a year ago, the U.S. Food and Drug Administration's Complex Innovative Trial Design Pilot Meeting Program, Progress to Date. So that brings us to challenge question number 2. This one is a true or false. True or false? The FDA has accepted Bayesian and adaptive designs for phase 3 studies. So I will pause again for about 30 seconds. The answer is true. We've accepted these kinds of designs under the CID program and outside of the CID program. So in summary, with regard to the Estimand framework, FDA is encouraging adoption of the Estimand framework, especially for confirmatory trials. And the Estimand framework really helps to facilitate clear communication about trial objectives and analyses between sponsors and FDA. And in terms of the CID review program, the FDA is committed to promoting the appropriate use of complex innovative trial designs to improve drug development, and sponsors who have design proposals in this category can submit them for consideration for the CID meeting program. So I'm happy to take your questions, and thanks very much for your time and attention.
Thank you, John, for this informative and enlightening presentation. As we see more product applications that employ different novel statistical methodologies, it's helpful for product developers to understand what programs and what resources may be available to them out there. So we have some questions here. Let me start with the first question. John, you spoke about the ICE strategy, and so this is a combined question. The first question is, how much of the ICE strategy needs to be specified within the protocol? And then the follow-up question, can elements of the ICE and certain testing strategies be provided within the statistical analysis plan instead? Thank you.
Thanks, Larissa. Great question and very useful for the audience. My most general advice on this would be to discuss with the FDA review division how they would prefer these things to be presented. The Estimand should be presented in the protocol and should be presented in more detail in the SAP. In terms of exactly how much detail goes into the protocol, we don't have very specific rules about that. But I would let you know that there's an additional ICH effort underway called M11, which is developing a clinical protocol template that will include guidelines on how to specify estimands in a clinical trial protocol and how much detail it should go into. So when that document is finalized, hopefully that'll provide some more specific guidance. Thanks.
Thank you, John. That's helpful to know. Our next question is, are multi-component endpoints generally considered to be under the category of innovative design? Thank you.
Sure. Thanks for the question. So the use of a multi-component endpoint in and of itself wouldn't usually be considered a complex innovative design. It could be one feature of a complex innovative design. But one thing to be aware of is another program at FDA, REdI, the Rare Disease Endpoint, I think, Assessment Program, which was just launched with PDUFA-VII, where for rare diseases, FDA is providing a structured meeting program to discuss the use of novel endpoints. So if you have a novel multi-component endpoint and it's a rare disease, you may want to consider applying to that program for discussion.
Thank you, John. And then the next question that we have here is, do you think this could be useful for a trial in amyotrophic lateral sclerosis? And I would probably ask to speak about all three components that we've discussed just now, multi-component endpoints, and an application to Estimand framework and the complex innovative design, because it would be helpful to know about all three. Thank you.
Oh, sure. Yeah, also a good question. I would think that the use of complex innovative designs in ALS could very well be a possibility. It's a very difficult development area, and thinking creatively about novel ways to approach clinical trial design and analysis, including adaptive designs and other methods, would be welcome. And I think the review divisions would welcome discussion on those topics. The Estimand framework we would definitely encourage for any late phase study in ALS or in any disease. We're recommending the use of the Estimand framework across the board, really, in confirmatory trials. And then in terms of multi-component endpoints, I'm not really a specialist in ALS, but I do think that typically assessing functional outcomes does involve looking at various aspects of symptoms and progression. So I think the use of multi-component endpoints is worth doing in ALS. Thanks.
Thank you. Thanks for covering all three. Quite informative. Another question that we have here is the following. Are estimands just for phase 3 studies, or are they required for early phase studies as well?
Also a good question, thanks. So we're recommending that people use the Estimand framework for all phase 3 studies and any other study that's intended to support substantial evidence of effectiveness for approval. The Estimand framework can be applied more broadly than that, and we certainly encourage sponsors to consider using it for phase 1, phase 2 studies, and potentially also studies outside of the clinical trial framework. There's a lot of interest in applying the Estimand framework to observational studies, for example. So our strong recommendation is yes for confirmatory studies and for hypothesis generating or exploratory studies. I think it's worth considering.
Thank you. Are you ready for the next question?
Absolutely, thanks.
So how does the Estimand framework work for per protocol analysis, for example, in non-inferiority studies? Thank you.
Thanks. This is a question that comes up fairly often because historically per protocol analyses have been used very widely as supportive or sensitivity analyses, and in some cases as primary study analyses. The Estimand framework described in ICHE 9R1 does not encourage the use of per protocol analyses in most cases, because in most cases they don't correspond to a coherent Estimand strategy. In particular, people sometimes think of per protocol as a version of a principal stratification strategy where you would be looking at subjects who were able to comply with the protocol as your population of interest. The difficulty is that in parallel arm studies, you don't observe subjects on both treatments in the study, so you don't know what would have happened in terms of their ability to comply with study procedures had they been assigned to the other treatment arm. There's some thought that in crossover studies, you may be able to use per protocol approaches as a kind of principal stratum approach, but in parallel arm studies, we don't recommend it. So this does cause conflict with the traditional approach in non-inferiority studies of using per protocol analyses as a way to work against the bias that can occur in non-inferiority studies toward finding two treatments to be comparable if there's high variability in the study of various kinds. What's recommended in ICHE 9R1, instead of kind of automatically using a per protocol analysis, the guidance recommends that people think very carefully about which intercurrent events might cause bias and what would be the most appropriate way of minimizing those events and of addressing them analytically when they occur. Thanks.
Great. Thank you, John. Our next question is this. Is intent to treat the same thing as a treatment policy estimate? Thank you.
Intent to treat and treatment policy strategies are very closely related. The way we would usually say it is that the use of a treatment policy strategy is aligned with the intent to treat principle. And people who are familiar with how studies are analyzed will sort of pick up on the fact that the way we would do an intent to treat analysis is essentially the same as applying a treatment policy strategy to an intercurrent event, which is to say that we analyze the outcomes of the patients regardless of whether the intercurrent event has happened or not. Patients are simply analyzed as randomized using all of their available data. Thanks.
Thank you. Our next question is about Bayesian trials. John, you discussed Bayesian design during your presentation. Does FDA accept Bayesian trials outside of the CID paired meeting program? Thank you.
Thank you, and thanks for the question. Yes, the short answer is yes. FDA is happy to consider any appropriate statistical methodology for clinical trials, Bayesian or frequentist, within or outside of the CID paired meeting program. The CID paired meeting program can be helpful for getting more intense feedback on complex designs, but FDA will consider Bayesian proposals outside of the program as well.
Thank you, John. Well, now let's leverage your expertise and experience in reviewing many protocols and statistical analysis plans. So the next question is about what has been used for approval, and specifically, has a Bayesian trial ever been used for approval of a new product? Thank you.
Thanks for the question. Yes, so Bayesian approaches have been used for certain aspects of approval for several products over the years, but in particular, in terms of kind of a primary Bayesian analysis and a Bayesian trial design, last year, I think last fall, CBER approved the first fecal microbiota transplant product called REbiota, based on a Bayesian phase 3 study that borrowed information from phase 2. So as far as I know, that was FDA's first approval with a Bayesian framework that used borrowing as the primary analysis. Prior to that, FDA approved the mRNA COVID-19 vaccine Comirnaty based on a Bayesian analysis, but that analysis didn't involve borrowing data. It was more of a kind of a light Bayesian approach. Thanks.
Thank you, John, for sharing these examples. Another question: how difficult is it for sponsors and the FDA to agree on disclosure for the CID meeting program? You did talk about disclosures within the framework of the complex innovative design program. So if you could please speak about it. Thank you.
Thank you, and thanks for the question. So yeah, FDA and a sponsor agreeing on which study elements can be publicly disclosed following entrance into the CID paired meeting program is a critical aspect of the program. And I'll say that when we were developing the program, we had some concern about whether this would be kind of a stumbling point for applications to the program. As it's turned out, this hasn't really been a stumbling point. We've had very productive discussions with each sponsor in the program and have been able to come to agreements, I would say, quite easily. I think both FDA and the sponsors are motivated to see increased use of appropriate complex designs, and it's been a really good collaborative process.
Thank you, John. And one more question about the CID meeting program. Is selection for this program, the Complex Innovative Design Meeting Program, competitive? Thank you.
Thanks. We wouldn't describe it as competitive. FDA will accept up to 2 applications to the program per quarter. In theory, we could have more than 2 in a given quarter and would have to decide between them. But in practice, most quarters we have selected fewer than 2. And so if you're considering applying to the program, I would definitely encourage you to do it. We would be really interested in seeing new complex proposals. Thanks.
Thank you, John. And I believe this will conclude our session on Estimands and the Complex Innovative Design Program. Thank you very much for the presentation, and thanks to the audience for sending your questions. We are now going on a short break, and we will see you back at 1:35. Thank you. Welcome back, everyone. Our next session is about vaccine products. Coming out of the pandemic and after massive vaccination programs have been implemented, it was no surprise to us to see requests in the last year's evaluation forms to include vaccine-related topics in our REdI event, and so we did. And in a few moments, you will hear an overview of the post-marketing safety monitoring for vaccine products. And our presenter on this topic is Dr. Meghna Alimchandani, who serves as the Deputy Director of the Division of Pharmacovigilance in the Office of Biostatistics and Pharmacovigilance in the Center for Biologics. Her work is focused on pharmacovigilance planning and passive and active post-market safety surveillance for products regulated by the centers. She also oversees the design of post-marketing studies and risk evaluation and mitigation strategy programs, and she oversees pharmacovigilance reviews for biologics license applications. Dr. Alimchandani joined FDA in 2014 and served in various review and supervisory roles on a variety of biological product programs. Prior to her work at FDA, Meghna received her medical degree from the Albert Einstein College of Medicine in New York and completed her residency in anatomic pathology at the National Institutes of Health. Please join me in welcoming Dr. Alimchandani, who will start her presentation in a few moments. Thank you.
I want to welcome everyone to this session on post-marketing safety and pharmacovigilance for vaccines. My name is Meghna Alimchandani. I'm from the Division of Pharmacovigilance within the Office of Biostatistics and Pharmacovigilance in CBER. Reviewers in my division conduct post-market safety monitoring for all CBER-regulated products, including vaccines, which is the subject of this presentation. My goal is to provide you with an overview of post-marketing safety monitoring for vaccines, including some examples of pharmacovigilance activities, such as safety-related post-marketing studies. These are the learning objectives for this presentation. We will describe FDA's regulatory responsibilities during product lifecycle with the focus on the post-marketing period. Pharmacovigilance is the term that we use to describe safety monitoring in the post-licensure period. Both passive and active surveillance are leveraged for this purpose. We will describe signal evaluation and risk management regulatory actions. I will provide a few examples demonstrating signal detection, evaluation, and risk management regulatory action, and we will end with summary and conclusions. Overview of FDA responsibilities during product lifecycle. The point of this figure is to show vaccine safety continues throughout the life cycle of the product, and there are many different activities performed by a multidisciplinary review team. The data from the clinical development program, both preclinical data and pre-market clinical trial data, inform the pharmacovigilance plan and proposed actions for safety concerns. FDA performs continuous vaccine safety monitoring throughout the product lifecycle. This starts with the clinical development program and goes on through the post-licensure period. Listed here are some examples of vaccine safety activities. Preclinical research of candidate vaccines, early phase human studies followed by large phase 3 clinical trials, inspection of manufacturing facilities, monitoring lot release, post-marketing adverse event surveillance, which is the focus of this session today, inspection of sites for compliance with GCP, etc. To reiterate, monitoring vaccine safety is equally important during the development as well as during the post-marketing period. Why does CBER conduct post-marketing vaccine safety surveillance? So post-marketing safety surveillance is mandated by the regulations for all licensed vaccines. Here we list some of the limitations of a pre-market safety database, which makes post-marketing surveillance critical to ensure vaccine safety. Clinical trials may not detect safety issues that arise when products are marketed to the general population. For example, post-marketing surveillance may reveal interactions with comorbidities. Inclusion criteria may exclude groups within the general population. For example, pregnant and lactating women are often excluded. Smaller sample sizes and observation periods limit reliable detection to the most common events with shorter latency to onset. Therefore, we may not see rare adverse events or adverse events with long latency in the pre-market database. Unless a trial has a dedicated safety endpoint, inferences about safety are limited by concerns about post-hoc analysis with multiple comparisons. As you know, in general, a clinical trial is powered for the efficacy outcome. Of course, the trial will collect data on adverse events and safety data, but it may not be powered to detect a specific safety outcome. A post-marketing targeted safety study, on the other hand, as we will describe later in the presentation, is powered to assess a specific safety concern. And sometimes this means that the post-marketing study will need a very large sample size to be adequately powered to detect a rare safety concern. Thus, it is during post-marketing surveillance that we are able to further characterize the safety profile of licensed vaccines. Pharmacovigilance for vaccines. I want to spend a few minutes on this slide. It's a busy slide and I will walk you through it. So this slide provides an overview of the key decisions in pharmacovigilance planning. Pharmacovigilance is defined by the World Health Organization, WHO, as the science and activities relating to the detection, assessment, understanding, and prevention of adverse effects or any other drug-related problems. Note that this is a broad definition and would include things like medication errors that may not be associated with the clinical adverse event. The pharmacovigilance plan contains the sponsor's assessment of safety specifications for their product based on the clinical safety database. The manufacturer proposes important identified risks, important potential risks, and important missing information. This safety specification is intended to help industry and regulators to identify any need for specific data collection and surveillance activities. Our job at FDA is to review the safety data and proposed surveillance activities and make a decision about the adequacy of the proposed pharmacovigilance actions. So the items listed on the right side of this slide describes surveillance activities. All pharmacovigilance plans include routine pharmacovigilance. Routine PV is mandated by the regulations for all licensed products and includes mandatory adverse event reporting by the manufacturer and submission of periodic safety reports by the manufacturer. Now, depending on the safety profile, certain products may have additional pharmacovigilance activities. There may be safety post-marketing studies. These may be PMRs or PMCs. We will talk more about post-marketing requirements and commitments. And finally, FDA has the regulatory authority to require risk mitigation under a risk evaluation and mitigation strategies REMS program to ensure that the benefits of a product outweigh its risks. At the bottom of the slide, I have listed FDA guidance documents for your reference. Our next topic is to describe passive surveillance for vaccines. Adverse event reports for vaccines are submitted to the Vaccine Pharmacovigilance Database, which is known as Vaccine Adverse Event Reporting System, or VAERS. VAERS was established in 1990, and it is co-managed by FDA and the Centers for Disease Control and Prevention, CDC. Okay. So with passive surveillance, we are able to conduct continuous safety monitoring for vaccines during the post-licensure period. VAERS serves as the nation's early warning system to detect possible safety issues with vaccines. VAERS is a passive reporting system containing reports of spontaneous adverse events, meaning it relies on individuals to report their adverse events after vaccination, and there is no solicited or active follow-up of these vaccinees for adverse events. Now, there are mandatory reporting regulations for manufacturers. All serious and unlabeled adverse events have to be reported within 15 days by the manufacturer. All other adverse events have to be submitted as non-expedited reports. The manufacturer also submits periodic safety reports, which contains aggregate review of safety data from different sources, and this may include foreign experience if applicable, as well as literature reports and the sponsor's assessment for any safety signals from any source that were identified in the reporting interval. Periodic safety reports may be submitted in different formats. For example, periodic adverse experience reports, PAERS, periodic safety update reports, or PSURs, or the periodic benefit risk assessment report, or PBRR. For the first 3 years post-licensure, the manufacturer submits periodic safety reports at quarterly intervals, followed by annual reports thereafter. It is also important to remember that in addition to VAERS, we have access to global pharmacovigilance resources. There are global databases maintained by the World Health Organization. Other foreign regulators have their own pharmacovigilance databases. FDA routinely exchanges information on potential post-marketing safety issues with foreign regulators. This figure illustrates the sources of VAERS reports. Patients, healthcare providers, family members submit reports to VAERS. Vaccine manufacturers submit adverse event reports to VAERS as per mandated adverse event reporting timelines that were described earlier. VAERS data is reviewed by both FDA and CDC. As I mentioned, VAERS is co-managed by FDA and CDC. We collaborate very closely with our CDC colleagues. I also wanted to note that VAERS data sets are redacted and available to the public via the websites that are shown in this figure. This is what the VAERS website looks like. Vaccine adverse event reports can be submitted online or via a downloadable PDF to VAERS. And again, we have the CDC Wonder data set that is publicly available. This figure depicts the temporal association from vaccination to adverse event. Note that there are no deadlines for the submission of a VAERS report, and that part of the timeline could really vary. But reports should be submitted promptly after an adverse event occurs to facilitate surveillance and review. This is an important slide listing the strengths and limitations of a passive surveillance database such as VAERS. VAERS is our early warning system for vaccine safety and has many strengths. Rapidly detects potential safety problems, potential detection of rare adverse events. It is open-ended for hypothesis generation, captures geographic diversity, and may have the capability to monitor product lots if that information is available on the spontaneous reports. Now on the right side are important limitations of a passive surveillance database. These limitations are very important to keep in mind when evaluating various data. There may be missing and/or inaccurate data in the reports. Reported diagnoses, this is very important, reported diagnoses are not verified. VAERS accepts all reports regardless of the plausibility of the vaccine causing the event or the clinical seriousness of the event. We know that there is underreporting with passive surveillance systems. There may also be reporting bias, such as stimulated reporting, that may be present. And keep in mind that an unvaccinated control group for comparison is not available. Due to these limitations, we may not be able to assess causal association. Because this is a passive surveillance system and depends on spontaneous reports, it is not likely to detect long latency events. To provide context for VAERS data, we refer to product utilization data or patient exposure data as available. We may also ask the sponsor to provide lot-specific distribution data when performing lot analysis of adverse events. Note that usually actual vaccine administration data is not available. A notable exception, though, was that during the COVID-19 PHE, CDC tracked vaccine administration data for the COVID-19 vaccines. But usually this level of data is not available, and we instead depend on product distributed in the US to give us a sense of denominator data. We have to keep in mind that we cannot calculate incidence rates. Not all doses distributed were actually administered to individuals, and distribution data is usually not available by age groups. Brings us to our first challenge question. Routine pharmacovigilance for vaccines includes A. Adverse event reporting in accordance with 21 CFR 600.80. B. Submission of expedited 15-day reports for serious and unexpected adverse events. C. Submission of non-expedited reports for serious unexpected and non-serious adverse events. D. Submission of periodic safety reports at quarterly intervals for 3 years post-approval and at annual intervals thereafter. E. All of the above. Okay, and the right answer is all of the above. These are all part of routine pharmacovigilance for vaccines. Okay, so our next topic is active surveillance activities as part of vaccine pharmacovigilance, and I have also provided the link to a guidance document on pharmacovigilance and epi studies for reference. Safety-related post-marketing study. We know that all studies and clinical trials collect safety data, but for the next few slides, when I refer to safety-related post-marketing study, I am talking specifically about a study being conducted to evaluate safety or further investigate a safety issue. In other words, a study must have a primary safety endpoint to be considered a safety-related study. The designs of these studies vary. Examples of safety-related studies include pregnancy registries, surveys, observational epidemiology studies, and studies using healthcare claims and/or electronic health records, EHR data, from population-based sources. There are two big categories of post-approval safety studies. Studies conducted by the sponsor or applicant or manufacturer. I'm using these terms interchangeably. So studies conducted by the sponsor, that's one category. And then the other category includes studies conducted by other stakeholders, such as U.S. government or academia or other organizations. In the category of sponsor studies, there are three types of sponsor studies: post-marketing requirements, post-marketing commitments, and voluntary sponsor studies. For safety PMRs and PMCs, our review at FDA includes review of study protocols, interim reports, final study reports. For protocol reviews, we provide comments to the sponsor regarding the study design, objectives, data source, study feasibility, sample size, if the study is adequately powered for the safety concern to be evaluated, and also the timeline for conducting the study. FDA closely tracks study milestone dates for final protocol submission date, study completion date, and the final study report submission date. The sponsor is mandated to submit annual PMR-PMC status update reports, which are reviewed by FDA. And the study status of these safety PMRs and PMCs are posted publicly. At the bottom of the slide is a link to the public website where anyone can look up study status for a safety PMR or PMC. Is it ongoing? Is it delayed? Is it completed? Etc. FDA also reviews any submissions for voluntary studies that are submitted by the sponsor and provides responses to any questions that the sponsor may have for us. But we do not track study milestone dates for the voluntary sponsor studies. I have a few examples of the other studies on the next slide. So these are some examples of non-sponsor vaccine active surveillance activities. The FDA Amendments Act of 2007, FADA of 2007, required FDA to develop a post-marketing risk identification and analysis system, and this enables FDA to access existing automated healthcare data by partnering with data holders such as insurance companies with large claims databases and owners of electronic health records. It strengthens FDA's ability to monitor post-marketing performance of products and augments existing systems. The CBER Biologics Effectiveness and Safety BEST initiative is engaged in active surveillance activities for vaccines. The protocols for BEST studies are posted on their website. We also leverage data from CMS, the Centers for Medicare and Medicaid Services, to conduct vaccine studies. And then I want to take a couple of minutes to describe the Vaccine Safety Data Link, or the VSD. The VSD is a collaborative project between CDC's Immunization Safety Office and integrated healthcare organizations and networks across the U.S. As you can see from this map, there are so many collaborating centers in the VSD network. The VSD started in 1990, and as of September of last year, September 2022, there are 13 VSD sites that provide clinical, methodological, and data expertise. The VSD uses electronic health data from participating sites to monitor and assess the safety of vaccines. These slides provide some more background on the difference between post-marketing requirements, PMRs, and post-marketing commitments, PMCs. Post-marketing requirements are, as the name implies, required studies. PMRs are for targeted safety studies to assess a specific serious risk. FDA has regulatory authority for enforcement. As I mentioned before, we closely track the status of PMRs and study progress. For any delays in study milestone dates, the applicant needs to submit a good cause request to provide their justification to support the delay, and this will be reviewed by the FDA. Post-marketing commitments, on the other hand, are defined as agreed upon studies between FDA and the applicant, and we do not have the regulatory authority for enforcement that we do with the PMRs. PMCs are usually general safety surveillance studies or for adverse events of special interest that do not meet the criteria for study purposes under FDA. And I'm going to describe these study purposes on the next slide. Okay, so post-marketing requirement is to assess a serious risk. This slide provides a reference to the FDA guidance document and lays out the study purposes under the regulations. Post-marketing studies and clinical trials may be required for any of the following purposes: to assess a known serious risk, signals of a serious risk, unexpected serious risk when available data indicate the potential for a serious risk. So here are some regulatory considerations for a safety PMR. Post-marketing studies and clinical trials may be required again under those FDA purposes that I just described on the previous slide. Before a PMR can be imposed on the sponsor, FDA must find that adverse event reporting, so spontaneous adverse event reporting, and the active post-marketing risk identification and analysis system as available under the FD&C Act will not be sufficient to meet the above purposes. Similarly, before requiring a post-marketing requirement clinical trial, FDA must first find that a study will not be sufficient to achieve the same purposes. The definitions for clinical trial and clinical study are further described in the FDA guidance document listed at the bottom of the slide. So now we come to two more challenge questions. Okay. Safety-related post-market study may include A, a post-marketing requirement study under FDA, B, an agreed-upon post-marketing commitment study, C, a voluntary sponsor study, D, an active surveillance study using population-based data sources, E, all of the above. Okay, and the right answer is E, all of the above. Safety-related studies include any of these. They could be PMRs, PMCs, voluntary studies, other studies being conducted by other stakeholders, different study designs, whether it's observational, prospective cohort study, whether it's a study using population-based data sources. All of this falls in the broad category, may fall in the broad category of safety-related post-market studies. Okay, challenge question 3. FDA tracks study milestone dates for the following: A, post-marketing requirement studies under Section 5050 of the FDCA, B, post-marketing commitment studies under Section 506B of the FDCA, C, voluntary sponsor studies. D, A, and B. Okay. Give you a few moments to think about this. So the correct answer is A and B. So for both PMRs and PMCs, 506B PMCs, FDA tracks the study milestone dates, and as I mentioned before, the status of the study is updated based on the annual status update reports and comparing with the milestone dates. And this information on the studies, on the status are available. This information is available on the website that I shared the link for earlier. FDA does not track the voluntary sponsor studies, and those are not listed on the approval letter either. So our next topic is signal evaluation and risk management. And for reference, this is a best practices document that provides a very good summary of our processes with regards to post-marketing safety surveillance. So post-marketing safety issue is defined very broadly as information from one or multiple sources which suggests a new potentially causal association or a new aspect of a known association that is judged to be of sufficient likelihood to justify verificatory action. This is actually modeled after the SIOMS definition for safety signal. Okay, the SIOMS is the Council for International Organizations of Medical Sciences. And note, I want to note that this broad definition includes medication errors or product quality issues that may not be associated with the clinical event. So this is a stepwise depiction of the entire process, all the way from signal detection to evaluation, verification, and risk management and regulatory action. So at the top, you can see there are many sources of safety data, okay, including clinical trial data, pre-licensure as well as post-licensure data, spontaneous reports, information from population-based data sources, other studies, literature reports, foreign regulatory agencies, etc. Okay, and with the review of all of this data helps us in detecting signals and to further evaluate signals. For signal verification, note that we may need additional studies. And finally, signal detection, evaluation, verification, all of this culminates in risk management and regulatory actions, such as communications, label changes, recalls, etc. The next couple of slides will touch on qualitative and quantitative analysis for passive surveillance data. Okay, so analysis of passive surveillance with qualitative methods. Some of the aspects of qualitative review at the level of the ICSR, or Individual Case Safety Report, includes looking for unusual clustering, trends in adverse events, biological plausibility, absence of alternative explanations, temporal association, and a series of cases for the same product or product type similar in the reported event and fitting a case definition for the event becomes a case series. Demographic and clinical patterns from case series can be analyzed and reviewed to further evaluate the potential association between the adverse event and the product. Quantitative review includes data mining and screening for disproportional reporting, which is further discussed on the next slide. Reporting rate is the number of reports received over a given time period. Reporting rate can also be expressed by the number of reports over the number of doses of a product. Reporting rates may be compared to background rates for an adverse event of special interest. Reporting rates are not an incidence. We must keep in mind that passive surveillance is limited by significant underreporting. So actual occurrence of an event is much higher than the reporting rate. Reporting rate can also be biased by the age of the product, the time that it has been used, that it is being used commercially, and the public interest in the adverse event, the seriousness of the event, and many other factors. Data mining helps us assess the proportion of reports with a specific vaccine adverse event pair compared to the background database of vaccine event pairs. This graph shows adverse events reported for a specific product with high statistical scores, alerting staff that further investigation may be warranted. Very important to keep in mind, an elevated score does not mean there is a causal association between a product and event. Data mining is a screening tool, and data mining alerts are hypothesis generating and are further evaluated. If a data mining score is elevated, these are some of the questions we ask ourselves. Is the adverse event unexpected? For instance, it's unlabeled. Is it confounded by indication? We may undertake additional case series analysis or epidemiologic studies. Risk management. Risk management and regulatory actions for safety signals are listed on this slide. One of the most important ways to communicate safety information about a product is through the approved US package insert. FDA has the authority to require sponsors to make safety labeling changes if the safety signal meets criteria for new safety information. Other than mandated safety labeling change, we also have numerous labeling supplements that are submitted voluntarily by the sponsor to make updates to section of the USPI. Other examples of risk management actions include safety communications, inspections, product withdrawal, recall, product and post-marketing requirement and commitment studies that we have discussed in detail previously, additional surveillance activities, additional active surveillance studies, and of course we can always publish data and present at professional meetings. So this brings us to our last challenge question for today. FDA has the authority to require safety-related post-marketing studies or clinical trials and to require safety labeling changes. True or false? Okay, and as we have seen, this is true. Indeed, FDA has the authority to impose post-marketing requirement studies. Okay, these are PMRs, and yes, we have the authority to require safety labeling changes if it meets certain criteria for new safety information. Okay, so in the next few slides, we will go over some examples to illustrate the concepts we have covered so far regarding post-marketing safety and pharmacovigilance for vaccines. So the next three slides present the example of Guillain-Barré syndrome, or GBS, after the Janssen COVID-19 vaccine. This was identified from various data. This analysis was published in the JAMA article that is cited here, and we also presented this at the ACIP meeting. And subsequently, we had a label change to add a warning for GBS. So you can see how the risk of GBS after this vaccine was communicated in multiple ways: ACIP meeting, vaccine safety label change, and publication. This is a busy slide, but it's a good example that shows observed to expected analysis and how that helps with signal identification. This is excerpted from the JAMA publication that was cited earlier. What it is showing you is an observed to expected analysis of GBS after the Janssen COVID-19 vaccine. Observed data is based on the GBS reports to VAERS. It is very, very important to note that this analysis was based on unconfirmed cases of GBS. Because this was based on passive surveillance data, there was incomplete or missing data in spontaneous reports that precluded confirmation of cases using Brighton criteria. So that's the observed cases. The number of expected cases are calculated based on background rates from published literature and age-specific dose administration data that was obtained from CDC. Observed to expected is calculated across different age groups. We know that the incidence of GBS increases with age. In the table, the results using the highest published background rates for each age group were used to illustrate the lowest OE ratio representing the most conservative estimate of the potential association. And on the rightmost column, you have rate ratios that demonstrated the increased risk with this vaccine. And this slide shows the addition of the GBS warning to the label. Okay, this is another example of safety signal identification from post-marketing safety data. Now, in the previous example, I showed you a signal for GBS that was identified from passive surveillance VAERS data. In this example, on this slide, we have a signal for GBS that was identified for Shingrix, and this time the signal was detected using data from an active surveillance study in CMS. This was an observational study of Medicare beneficiaries that demonstrated an increased risk of GBS during the 42 days following vaccination with Shingrix. Of note, these analyses of GBS, unlike my previous example, these analyses of GBS diagnosis and claims data were indeed supported by analysis of GBS cases confirmed by medical record review. FDA required a warning for GBS. This was a required, mandated safety labeling change, and a safety communication was posted on the CBER webpage that is cited here. And the results of the study were also published. So again, you have multiple ways that this risk was communicated. Mandated safety labeling change, safety communication on the webpage, peer-reviewed publication. This is another example of safety signal identification from a post-marketing study conducted by FDA. This is an older example from 2013. The RotaTeq label was updated with the findings from the study regarding the risk of intussusception after RotaTeq. The risks of intussusception must be considered in light of the demonstrated benefits of rotavirus vaccination. Again, in this case, you have multiple channels of risk communication by the FDA. Safety labeling change, safety communication, peer-reviewed publication. As I mentioned earlier, VAERS is co-managed by FDA and CDC. CDC Immunization Safety Office reviews the VAERS data. Okay, and over here are two websites. These are examples of CDC web pages containing information for the public regarding vaccine safety. The first website is for influenza vaccines, and the second one is a screenshot of the web page for selected events reported after COVID-19 vaccines. The next two slides are examples of another forum for risk communication and discussion of safety data with the public. FDA presenting data at advisory committee meetings. For vaccines with a pediatric indication, FDA presents our review of comprehensive post-marketing safety data from spontaneous reports, manufacturer periodic safety reports, and published literature to the Pediatric Advisory Committee, or PAC. We present vaccine data at the VRBPAC, the Vaccines and Related Biological Products Advisory Committee meetings. Earlier this year, we had VRBPACs for RSV vaccines. There was much discussion about safety profiles for these novel vaccines and discussion of the pharmacovigilance plans and additional active surveillance studies to assess certain risks. In the last few years, the VRBPAC met frequently to discuss data on COVID-19 vaccines. This slide shows some examples of post-marketing safety studies. RXV is the first respiratory syncytial virus, RSV vaccine, approved in the US. It was approved earlier this year in May. Post-marketing requirements and commitments along with their study milestone dates are listed in the approval letters that are posted publicly on FDA web pages. On the left of the slide is an example of a post-marketing commitment for atrial fibrillation. On the right side of the slide is an example of a safety PMR to assess signals of the serious risks of GBS and ATEM, acute disseminated encephalomyelitis. Okay, and this is all again excerpted from the approval letter for RXV. Summary and conclusions. So pharmacovigilance review is based on safety data and signal detection from pre-market data. We review the sponsor's proposed pharmacovigilance plan. The PVP is a living document, continues to be updated based on accumulating post-marketing experience and based on any signals that may be detected from post-marketing data. Signals from pre or post-licensure data informs our plans for post-marketing studies and or additional surveillance activities. So in summary, despite rigorous safety evaluation during pre-market phases of clinical development, post-marketing safety monitoring is necessary due to limitations of clinical trials. FDA conducts continuous safety monitoring of all licensed vaccines. Post-marketing safety monitoring includes passive and active surveillance. FDA may require post-marketing studies by manufacturers. FDA may require safety labeling changes. And then new databases have expanded population-based surveillance capabilities. We are increasingly leveraging claims-based data and AHR data for post-marketing studies. FDA and CDC are in close collaboration and share many vaccine surveillance activities. And finally, our goal is to ensure safe and effective vaccines. Thank you so much for your attention. This is the end of my presentation, and I'm happy to take questions. Thank you so much.
Thank you very much, Meghna, for this presentation. It's good to know what to expect after vaccine products are approved and how their safety continues to be monitored and assessed. And we're now at the question and answer part of the session, and I see quite a number of questions here in the chat box. Not surprising that there is interest on this topic. And let me start with the first question. And here it is. Meghna, you presented on post-marketing safety monitoring for approved vaccines. How does this compare to post-authorization safety monitoring for the COVID-19 vaccines under the EUA? Do the requirements for AE reporting under CFR 680 apply to vaccines under emergency use authorization? Thank you.
Hi, thanks, Larissa. So I wanted to quickly compare the adverse event reporting for a licensed vaccine versus an authorized vaccine. So as we went over in this talk, the adverse event reporting for an approved vaccine, so licensed vaccine that was approved under a BLA, Biologics license application, the mandates for adverse event reporting come from the Code of Federal Regulations. But for an EUA, the mandate for adverse event reporting and all the reporting requirements are actually listed in the letter of authorization itself. So if you, all of these letters of authorization, for example, for the COVID-19 vaccines are available publicly on FDA web pages. And there is a section for conditions of authorization. And under those conditions of authorization, we have various reporting requirements, both for the manufacturer as well as for vaccination providers. We also have requirements for the manufacturer to conduct post-authorization safety studies. Thanks so much.
Thank you, Meghna. Our next question is a combined question, and it relates to the vaccine adverse event reporting system. So it contains a couple of sub-questions. How often does FDA review vaccine-related adverse events in VAERS? And what is the process that FDA follows to conduct the review? And there is a follow-up question here. How is the VAERS FDA review documented and communicated to the public?
Thanks for this question. So I think this question came in early on in the presentation. So hopefully after you listen to the talk, you know some of these answers already. So in terms of how often does FDA review vaccine-related adverse events in VAERS, it's really a continuous process, and it really depends for different vaccines. What are the ongoing safety issues? How old are the vaccines? Is it a flu vaccine that is really, influenza is a vaccine that is only going to be administered during the flu season? So we take all of that into consideration and have a risk-based approach. But really, we're doing continuous vaccine safety monitoring and using multiple tools. We're looking at the VAERS data. We're also getting information in periodic safety reports from manufacturers. We're also getting information from other global agencies and inquiries that we respond to. So in terms of what is the process, so I think I went over that. We use both qualitative review as well as quantitative review, and part of the qualitative review is really looking at individual case safety reports, doing case series analysis, looking at the adverse event and gleaning all the data about time to onset, when did the adverse event happen in relation to the vaccination, were there other concomitant vaccines, what is the biological plausibility, asking ourselves all those questions. And in terms of quantitative review, we're looking at data mining alerts, we're looking at observed to expected analysis. So we're comparing sometimes with background rates for certain adverse events. We're taking all of that into consideration. And then I think the last question was how is the VAERS FDA review documented and communicated to the public? So as I went over in the slides, we have numerous ways of communicating it to the public. If there is a safety signal, then that falls under risk communication. We can do safety communications on FDA web pages. We can also have label changes. That's our way to communicate to the public. It could be voluntary, depending on what the safety concern is. It could be a voluntary labeling change or a mandated, required safety labeling change. So that is another way that we're communicating risk to the public. We can have publications, and then for a review of VAERS data that is not associated with a safety signal, but we are just presenting our review. There are different ways that we do that as well. So we have different advisory committees. We have the VRPAC advisory committee. We have pediatric advisory committees where we provide a comprehensive summary of post-market safety data. So I hope that helps to answer the question.
Thanks, Meghna, for this very comprehensive response. One more question about VAERS. What is the difference between FDA's VAERS and CDC's Wonder databases? Thank you.
Yeah, that's a good question. So again, VAERS is co-managed between FDA and CDC. It's not, these are not separate entities. So VAERS is FDA and CDC. I would say it's the same, pharmacovigilance database that's co-managed. Wonder database is the public-facing data set of VAERS. That's all it is. The Wonder database is accessible to the public. Thanks.
Thanks, Meghna. Here's another question. Is there a mandatory request for providers or doctors to report the serious adverse events in a timely manner? Because providers would be the first line of notice. They would get these reports or occurrences sooner than the manufacturers will find out about them. So the question is there a mandatory request for providers or doctors to report adverse events? Thank you.
Yeah, thanks for this question. So there is the timelines for reporting, the mandated timelines for reporting do not apply to non-manufacturer sources of reports. So really we are, as FDA, we are regulating the manufacturers. We're telling them that certain adverse events you have to report within this timeline and this timeline. And with as regards healthcare providers and the public, the reports are really voluntarily coming from them. FDA does not regulate those timelines, and I will, why, and as to the reason for why, I would say, FDA is not regulating clinical practice. That's not our job. But I will say most of the reports that healthcare providers submit to VAERS are indeed, timely. It's not that they are waiting, weeks and months to submit from the time to onset of the adverse event. So, I think that's the response. Thanks.
Thank you very much again, Meghna. And our timer tells us that we have to conclude the session. Thanks again to the conference participants and to both of our presenters this afternoon, Dr. Scott and Dr. Alimchandani, for their interesting presentations and for responding to multiple questions from the audience. We will now go on a 10-minute break, and after the break, we'll transition to the next session, which will be about expanded access to investigational therapies and will be moderated by Dr. Zehra Tosun. We will see you after the break, which will be at 2:40 Eastern Time. Thank you.
Good afternoon. My name is Zehra Tosun, and I will be your moderator for the next two sessions. Now, it is my pleasure to introduce our next speaker, Dr. Xu. Dr. Xu is the Chief of General Medicine Branch 2 in the Office of Clinical Evaluation within Office of Therapeutic Products in CBER. Her branch is responsible for reviewing clinical trial protocols and evaluating trial data of investigational biologics regulated at OTP in several clinical areas, including neurology, ophthalmology, dermatology, and burn and wound care. Her branch reviewed all the clinical data that led to FDA approval of several gene and cellular therapies. Some examples include the first adeno-associated virus vector-based gene therapy products, Luxturna and Zolgensma, and more recently, the first topical gene therapy, Vyjuvek, for dystrophic epidermolysis bullosa. In addition to the regulatory responsibilities, she's actively involved in FDA guidance development, and some examples include expedited programs for serious conditions, human gene therapy for retinal disorders, and human gene therapy for neurodegenerative diseases. Dr. Xu received her MD from Central South University, Xiangya School of Medicine in China, and her PhD in neuroscience from Yale University. She completed residency training in neurology at Loyola University in Chicago. She is board certified in neurology by the American Board of Psychiatry and Neurology. Dr. Xu's upcoming presentation will outline the regulatory requirements on expanded access to investigational drugs and biologics for treatment use and will provide practical recommendations on how to submit expanded access IND requests to FDA CBER. Now I will turn the microphone to Dr. Xu.
Thank you for the kind introduction. I hope my talk will help you understand FDA's regulatory and statutory requirements of expanded access to investigational drugs for treatment use. I also hope I would provide some responses to some of the common questions related to expanded access to investigational drugs for treatment use. Throughout my talk, the term investigational drugs refers to both human drugs and biological drugs products regulated by CDER and CBER. What is expanded access? Expanded access or treatment use is to use an investigational drug with the primary purpose is to diagnose, monitor, or treat a patient's disease or condition. It is in contrast to clinical studies of investigational drugs, whose primary purpose is to obtain information about the safety and effectiveness of the drug. Occasionally, you will hear the term compassionate use or pre-approval access. However, please note, neither term is defined or described in FDA regulations. For someone who is interested in the expanded access program, Subpart I of the 21 Code of Federal Regulation 312 is critical because it includes criteria that must be met to authorize expanded access use, the requirements for expanded access submission, as well as safeguards that are intended to protect patients and to preserve the ability to develop meaningful data about the safety and effectiveness of the drug through clinical trials or drug development. In 2016, the 21st Century Cures Act was passed. In that Cures Act, it amended the Food, Drug, and Cosmetic Act by requiring this expanded access policy. which is a requirement to the manufacturer or the distributor of an investigational drug to make its policy for evaluating and responding to expanded access requests public and readily available. For example, by posting the policy on a publicly available website to include the contact information, the procedures for submission of expanded access requests, the general criteria for evaluation and response, the anticipated time frame for acknowledgement of such requests, and a hyperlink or other references to the record in clinicaltrials.gov that contains information about availability of the drug under expanded access. In 2017, the FDA Reauthorization Act amended the Food, Drug, and Cosmetic Act again to require that the expanded access policy for an investigational drug be posted by the earlier of either the first initiation of a phase 2 or phase 3 study of that particular drug, or within 15 days after the drug receives a fast track, breakthrough, or regenerative medicine advanced therapy designation. So a few points I would like to clarify. Posting of the expanded access policy does not guarantee access to the investigational drug under expanded access. It is the sponsor's voluntary action to provide expanded access to its investigational drug. We at FDA have a long history of facilitating expanded access to investigational drugs for the treatment use of serious or life-threatening conditions lacking satisfactory therapeutic alternatives. However, we cannot compel a sponsor to provide expanded access to its investigational drug. So what types of regulatory submissions can be used to obtain expanded access to a drug? It can either be an expanded access protocol, which is a protocol amendment submitted to an existing IND, or it can be an expanded access IND, which is a separate and a distinct IND from any existing INDs, and it is intended only to make a drug available for the treatment use under expanded access program. So when should an expanded access protocol submission be used? It should be used only if the sponsor seeking expanded access has an existing IND in effect. Typically, a pharmaceutical company or manufacturer of the drug with an existing IND. And this company or the manufacturer is developing the drug for marketing under the IND. When there is an existing IND in effect, we generally prefer that the expanded access protocol be submitted under the same IND. So with everything in one place, it is much easier both from administrative perspective, from the review perspective, and from the less burdensomeness for sponsors and FDA perspective. When should a new expanded access IND submission be used? When there is no existing IND in effect, or even though there is an existing IND effect, but the sponsor is not intended to be the sponsor of the expanded access use. For example, for individual patient, single patient IND, the sponsor of the existing IND may prefer that a patient's physician be the sponsor investigator and submit a separate individual patient IND. How do we categorize and subcategorize expanded access submissions? The expanded access can be separated into 3 categories. One is for individual patient. This includes for emergency use. Second is for intermediate-sized patient population. And the third is for widespread treatment use. Under each category, this could be submitted either as an IND or as a protocol, as I discussed briefly earlier. Now I'm going to talk briefly about expanded access for individual patients. It is an expanded access to an investigational drug for treatment use by a patient. It can be submitted as an individual patient expanded access IND, also known as single patient IND. In this case, the submission will be treated as a new IND, and there is a 30-day period from the date we receive the IND prior to the initiation of the treatment. However, in many cases, we may allow treatment to begin earlier. Alternatively, this expanded access for individual patients can be submitted as an individual patient expanded access protocol, also known as single patient protocol. So in this case, the protocol is submitted as a protocol amendment to an existing IND by the sponsor of the existing IND. And there is no 30-day waiting period. As long as the IRB approval is obtained, the treatment can start. However, we may put the protocol on clinical hold if we identified some critical issues, for example, safety issues, which cannot be resolved through the interactive review process. Under the expanded access for individual patients, there is a subcategory, which is the expanded access for individual patients for emergency use. It is intended to provide expanded access in an emergency situation. For example, treating a patient before a written submission can be made, and the treatments are expected to have a rapid effect on an acute clinical emergency. Such request is often submitted and authorized by phone or by email. The treating physician, who is the sponsor, must agree to submit a written submission within 15 working days of the initial authorization. The treatment may start immediately upon our authorization. And meanwhile, the treating physician or the sponsor must report to the IRB within 5 working days of emergency use if no IRB review is obtained prior to treatment. Intermediate-sized patient population expanded access. It can be provided if we determine that there is enough evidence that the drug is safe at the dose and the duration proposed for expanded access to justify a clinical trial of the drug in the approximate number of the patients expected to receive the drug under expanded access, and at least the preliminary clinical evidence of effectiveness or of a plausible pharmacologic effect of the drug to make expanded access use a reasonable therapeutic option in the anticipated patient population. The number of the patients under the intermediate-sized patients is more than 1, but generally less than a treatment expanded access. IRB approval is required prior to the treatment. Expanded access to an investigational drug can be provided under a treatment IND or protocol only if the drug is being investigated in a controlled clinical trial under an IND designed to support a marketing application, or all clinical trials of the drug have been completed and the sponsor is actively pursuing with due diligence marketing approval of the drug. And again, IRB approval is required prior to the treatment. So what forms are used for expanded access submission? There are two kinds of forms. One is the form FDA 1571, which is the same form for the IND submission. So this form can be used for all three categories of expanded access programs. There is a unique form called FDA 3926. It is an individual patient expanded access IND form. So it may be used by the licensed physician acting as a sponsor investigator. The form is more streamlined for those physicians to submit an individual patient IND. So what information need to be included in the expanded access submission? So it has to contain all the information required by 21 CFR 312-305B. In addition, there are other informations required for each category of the expanded access as listed here under different sub parts of the 21 CFR 312. Are there safeguards in place for expanded access use of an unapproved drug? The answer is yes. There are multiple layers of safeguards. This includes FDA's oversight of patient safety, and also the investigator has several responsibilities to ensure the safety, including reporting adverse events to the sponsors, ensure that the informed consent document requirements are met, ensure an IRB review is done. And the sponsors also have several responsibilities listed here. They have to comply with the expedited IND safety reporting requirements, and they have to submit to us the annual report if the IND or the protocol continues for more than a year. They have to ensure that licensed physicians are qualified to administer the drug, and they need to provide licensed physicians with the information needed to minimize the risks. And they also need to maintain an effective IND for the expanded access use. So how do we address individual patients' expanded access applications for treatments with multiple courses of therapy or treatment of a chronic condition? So this is not uncommon, although generally we would like to limit this expanded access of a single patient to a single course of therapy for a specific duration. We may authorize multiple courses of therapy or chronic therapy, including authorizing individual patients expanded access to treat a chronic condition that requires extended treatment, when the circumstances of the treatment are well defined and reasonable, considering the available evidence to support such use. The patient's physician proposes the full courses of treatments when filing the request of the expanded access, and they have a good justification so that we can determine that the benefit is likely outweigh the risk in that situation. However, please be aware that we usually do not allow expanded access for an unspecified duration at the discretion of the treating physician. Is IRB review and approval required for all expanded access categories? The answer is yes, except that for emergency expanded access, when there is no sufficient time to secure prospective IRB review. In that case, IRB must be notified within 5 working days of the emergency use. For non-emergency single patient IND, we allow for waivers of the requirement for review and approval at a convened IRB meeting. For intermediate IND or protocol and the treatment IND and protocol, a full IRB review is required. And the Form FDA 1571 requires a commitment that an IRB will be responsible for the initial and continuing review of the studies under an IND. So in Form FDA 3926, remember this is a form specifically for a single patient IND when the sponsor is the treating physician. Under this, so there, the treating physician can request the IRB review by either the chairperson or by a designated IRB member, rather than by the full IRB committee. Expanded access submissions subject to the informed consent requirements. Expanded access to an investigational drug for treatment use, including emergency use, requires informed consent document as described in the 21 CFR Part 50, unless one of the exceptions listed there applies. The informed consent must contain information set out in the 5020 and 5025 to allow the patient to make an informed decision about receiving experimental treatment. And how do we determine that authorizing expanded access to a drug will not interfere with clinical trial or drug development? So the potential for expanded access to interfere with clinical trials or drug development is high for rare diseases due to limited number of subjects available. This potential is highest early in development and decreases as development continues. In general, for rare diseases, drug development, well-controlled clinical trials should be initiated before treating patients under expanded access. And expanded access should be sought only for patients truly not eligible for or are unable to participate in those well-controlled clinical studies. And we strongly encourage sponsor consider study designs that can help to minimize barriers to trial participation. For example, broaden the inclusion criteria, incorporate virtual or at-home visits, or using health facilities closer to those potential study participants. What are some of the reasons for FDA to deny a request? I believe we approve most of the single patient expanded access requests. However, in certain situations, we may deny some of those requests. Because even when there are 2 or more individual patients expanded requests, for patients with the same disease or condition, there may be significant differences in clinical presentation of the disease or condition that make the risks acceptable for one patient but not for another. A patient may be able to enroll in a clinical trial that was not accessible to a previous patient who was granted expanded access. Or FDA become becomes aware, since authorizing a previous request for expanded access, that expanded access is impeding the clinical development of the drug, and on that basis, place further requests for expanded access on clinical hold. So now I'm coming to challenging questions. Question 1. I'm going to read the question first. IRB review and approval is required for all expanded access prior to investigational drug treatment, except for emergency expanded access use when there is no sufficient time to secure prospective IRB review. So is this true or it's false? I will give a few minutes before moving to the answer. So yes, the answer is true. Although the IRB approval is required for all expanded access program, however, for the emergency use, the IRB approval can be obtained within 5 working days after the treatment. So question 2, which of the following statement is not true? A, posting the expanded access policy guarantees access to the investigational drug under expanded access. B, FDA has a long history of facilitating expanded access to investigational drugs for treatment use for patients with serious or immediately life-threatening disease or conditions lacking satisfactory therapeutic alternatives. C, FDA cannot compel a sponsor to provide its investigational drug for expanded access use. D, the primary purpose of expanded access is not to obtain information about the safety or effectiveness of a drug. Yes, the response is A. So although it is required that the drug manufacturer posting those policies online, but there's no guarantee that the investigational drug would be provided under the expanded access program. So in summary, expanded access is to use an investigational drug with the primary purpose of diagnosing, monitoring, or treating a patient's disease or condition. FDA has a long history of facilitating expanded access. However, it is the sponsor's voluntary action to provide expanded access to their investigational drug. FDA has oversight for the patient safety for those treated under the expanded access program. So here are some of the resources, including 3 guidance documents. One of them is a recently published, updated Q&A guidance regarding the expanded access program. And here is my contact information. Thank you for your time. I'm looking forward to more discussion during the Q&A session. Thank you.
Thank you, Dr. Xu, for your wonderful presentation. And I also would like to thank the audience for their participation. We received quite a few questions that are very interesting. Our first question to you: when should individual patient expanded access use the emergency procedure?
Thank you, Zara, and thank you for the question. So we consider it appropriate to require individual patient expanded access using this emergency procedure when the requester feel the treatment of the patient must occur within a very limited number of hours or must occur before the next business day after regular business hours. So, and we intend to authorize those type of single patient emergency IND only when the situation is a true emergency. So if we determine that the situation is not a true emergency but qualifies for an expanded access, we will accept it as a non-emergency individual patient IND once the application is officially submitted. So in such case, the sponsor can treat the patient when the IND goes into effect 30 days after we receive the submission, unless the IND is placed on clinical hold. Or if we have the resource to finish the review of the request, we may notify the sponsor before the 30-day due date. And as I mentioned during my talk earlier, the sponsor has to agree to submit an expanded access IND or protocol within 15 working days after we authorize the use of the investigational drug under the emergency IND situation. Thank you.
Thank you so much for your comprehensive response on this subject. And we have another question. So can you talk a little bit on what will be the roles of the patient's physician and FDA in determining if expanded access for an individual patient is appropriate?
Oh, sure. So I think a lot of times it is the patient's treating physician who would be the one to request a single patient expanded access. So in those scenarios, the physician, before they make that request, they must determine that the probable risk to the patient from this investigational drug is not greater than the probable risk from the disease or the condition that they intend to treat. So the physician should make this determination based on the information about the drug available to the physician and also their knowledge of the patient's clinical situation. We do understand that there is often limited information available to physicians about the risks and the benefits of an investigational drug. And for us, we have to determine based on information available to us that the potential benefits outweigh the potential risk for the use of the investigational drug for this particular patient, taking into consideration of their disease or conditions. I think you understand that we have access to considerably more information about the overall risk profile of the investigational drug than the patient's physicians. And also we understand much better than the potential benefit. Therefore, we may reach a different conclusion than the requesting physician based on the information we have about that particular drug. Unfortunately, in most cases, we'll not be able to share the information about the particular investigational drug with the sponsor. And in addition, we have to determine if the patient under consideration has a serious or life-threatening condition and has no other comparable or satisfactory therapeutic options. And we also have to determine whether providing the expanded access wouldn't interfere with the development of the drug for the expanded access use. And lastly, we also have to make sure that the patient cannot obtain the drug under another IND or protocol, for example, a research clinical IND. And so because we have to make those decisions, it will be very helpful for the requesting physicians to provide sufficient information, for example, the patient's condition, including their manifestations, their clinical exam findings, and to provide the list of the medications or other treatments which have been tried and their responses. And also all those would help us to make the determination that we need to decide whether it's appropriate to approve this expanded access. Thank you.
Thank you so much for your immediate feedback on this subject. We have another question, and I think you have touched some of these topics in the next question in your presentation too. So the question is asking, when it is appropriate to request expanded access for multiple patients using an intermediate-sized patient population expanded access IND or protocol, rather than a treatment IND or protocol?
All right, I'll be happy to give a try. So I think I will put this into two different scenarios. One is when the investigational drug is being developed for eventually to be marketed. And so in this case, the immediate size patient population expanded access, either an IND or protocol, is used earlier in the development than the treatment IND or protocol. And also, if we determine that the clinical development of the drug is essentially complete, for example, the clinical trial to support the marketing approval of the drug has ended, and the intent of the expanded access is to bridge the gap between the completion of the clinical trials and the marketing of the drug to ensure that treatment is not interrupted, or to expand the treatment to additional patients. So in this case, no matter how many patients are expected to be treated, as long as there are more than one, a treatment IND or protocol will be used. And the second scenario is if the drug is not being developed for marketing, and the expanded access IND or protocol is intended to treat more than one patient. So the expanded access would be provided on the intermediate size patient population expanded access IND or protocol. In this case, in this scenario, treatment IND or protocol is not appropriate, period. Thank you.
Thank you so much. Our next question is basically covering more this subject from an administrative perspective. So one of our audience is asking if Form 1571 and Form 3926 can be used interchangeably, and what is the difference between them?
Okay, so the Form 1571 is used for the submission of all different kinds of INDs, whether it's a research IND or it's an expanded access IND. And the Form 3926 is specifically designed to make the single patient expanded access more streamlined when the requester is a treating physician. So in that case, the treating physician will serve as the sponsor as well as the investigator for the treatment use of a particular investigational drug for a single patient, either on the emergency or non-emergency situation. So if you look at the Form 3926, it basically guides you through to fill out the information, for example, patient's current condition, the treatment plan, the monitoring plan, and even the IRB approval, whether you are okay with just have the IRB representative or the IRB chair instead of full IRB approval. So I think the 3926 is trying to give the physician who will be the investigator / sponsor more guidance to make sure the application goes through more efficiently and it's less burdensome. Thank you, that's it.
Thank you so much. And I have one last question, and this question is asking about a specific scenario. So the question is, during clinical development, if the investigator determines that the benefits of administering an out-of-specification autologous therapy to a patient outweighs the risks, is notifying the FDA and IRB acceptable, or a single patient protocol under the IND is mandatory?
I think my recommendation in this case is I assume this patient is enrolled on the investigational IND, for which you obtain the patient sample to manufacture the autologous product, which is out of spec. So I think probably it's a good idea to specify that in the clinical protocol, what should be done in this kind of scenario, whether this could be given as a protocol deviation so that the patient would still be followed under this study protocol, or whether this patient should be taken out from this protocol and be treated under a single patient expanded access protocol, but could still be under the same IND. I think it is important to specify those in the protocol because by the end of the day, I think when you analyze the data, it'd be helpful. If you don't see a good effect in this group of patients with out of spec, then you at least have some idea of why that happens, or potentially this could be one of the causes.
Thank you so much for your comprehensive response. So that was our last question for you in this session. Thank you so much for your very interesting presentation and providing responses to the audience questions. And I would also like to thank the audience for all those great questions that came in. Now our session, this session will conclude, and we will take a break until 3:30 p.m. And I will be back as your moderator, and we have one more presentation for you. Break at 3:30 p.m. in Eastern Standard Time. Thank you. Hi, welcome to the last CBER session. My name is Zehra Tosun. I will be the moderator of this session and now introduce the last speaker for today and also for this year's REdI 2023 conference. Our next speaker is going to be Dr. Wang. Dr. Wang is a microbiologist at FDA CBER at the Office of Compliance and Biologics and Quality and Division of Manufacturing and Product Quality. Dr. Wang has a bachelor's and master's degree in engineering of industrial microbiology from South China in the Institute of Technology and a PhD in biology from New York University. Dr. Wang has performed regulatory review of different types of submissions, including IND and BLA reviews, and also conducted pre-license and pre-approval inspections in support of the review of original BLAs and prior approval supplements. Dr. Wang's presentation will summarize the systems that are covered by the FDA pre-license inspection and pre-approval inspection of facilities for cell and gene therapy products, including the facility design qualifications, quality systems, and examples of objectionable observations. Now I will turn the microphone to Dr. Wang for her presentation.
I'm Wang. I would like to thank the meeting organizer for the invitation to present on topic CBER inspection of facilities for biological products. The topics of this presentation including a brief introduction of CBER inspections, including pre-license inspections and the pre-approved inspections. And we will discuss about the systems and the major elements covered during CBER inspections. And then we go through some examples of FDA Form 483 observations that are identified during CBER inspections of manufacturing facility of biological products. CBER inspections are conducted based on the applicable regulations, Public Health Service Act Section 351 and FD&C Act Section 704 and the various parts of Title 21 CFR. CBER inspections play an important role in review process. Based on Public Health Act and the CFR, biological license should not be issued except upon determination that the product and the establishment are complying with standards established in the BLA and the requirements prescribed in applicable regulations. Based on FD&C Act, an inspection, if needed, is considered to be a part of the complete review of an application. A PAI supports the review of a BLA. A PAI supports the review of a PAS. And if there are any 43 observations identified during CBER inspection, those 43 observations must be resolved before the approval of your application. FDA compliance inspections, including CBER inspections and FDA ORA surveillance inspections. CBER inspections are conducted by CBER DMPQ, performed around the middle cycle review clock of a submission. CBER inspection are always pre-announced because we want to observe the manufacturing process for a specific product. CBER DMPQ performs facility review at different levels. We perform high-level review of facility and equipment information in an IND meeting materials. We perform in-depth review of facility and equipment in your BLA submission or in a PAS submission. We verify the submitted information during the inspections. ORA surveillance inspection are conducted by FDA ORA and may include a product specialist from a product office. ORA inspection verify CGMP compliance of an entire manufacturing site, including all drug substance or drug products that are manufactured on that site. CBER inspection objectives include to verify the manufacturer's compliance with the CGMP requirement, and to verify accuracy and the completeness of information submitted in a specific submission, such as a BLA or PAS. To review documentations that is not included in submission, but is a part of the overall review of the product. To evaluate manufacturing control, and which we'll discuss more in this presentation. To observe critical manufacturing process, including aseptic processing and other sterility assurance or microbial quality steps. CBER inspection team includes a DMPQ leader inspector who often review the facility and the equipment information in the submission. We have product specialist from product office who often review the CMC and the product information in your submission. DMPQ invite ORA inspector to participate in the CBER inspection. Sometimes ORA will send their inspectors based on the availability of their inspectors. To evaluate if an inspection can be waived, we follow the CBER SOP 8410 to determine when a pre-license or pre-approved inspection is necessary. It's a risk-based assessment, and we will look at the FDA inspection history in the facility. Prior to CBER inspections, DMPQ will request a production schedule so that the inspection team can discuss and determine inspection date. Then we will have a teleconference with the sponsor or the facility to discuss logistic item, including inspection date and the actual inspection working hours and any safety requirements, availability of inspection meeting rooms and your subject matter experts. CBER will send a document request for a list of documents. Some of the documents should be made available prior to inspection based on FD&C Act 704. Those includes a list of deviations, index of all SOPs, certain documents and the records. Other documents should be made available on site for inspector in the first day of the inspection. Those including specific SOPs, for example, deviation, CAPA, change of control, changeover procedure, environment monitoring protocol and procedure. And we also request records reports for environmental performance qualification, and for equipment qualification records or reports, and for manufacturing batch records. Routine CBER inspection activities include an opening, a closing, and everything between the inspection daily activities. Opening is happening on the first day of inspection. During the opening meeting, the FDA leader inspector will make a brief introduction for the inspection. Then all inspectors will present their inspection credentials to the most responsible personnel of the facility, and we will issue a Form 482 for facilities that are located inside the United States. Then firm will make an introduction presentation and to introduce about your facility, your organization history, what the products are manufactured on the manufacturing site, and your quality, production, and different systems in your facility. After that, inspection team will conduct a walkthrough, also known as inspection of facility. We will start from raw material receiving and warehouse, and follow the material, and we'll go through the manufacturing process area, including upstream manufacturing facility, downstream manufacturing facility, and the drug product finish and filling facility. The inspection daily activities including review and the discussion of various SOPs and records with your SMEs. And we will observe manufacturing operations, which may or may not occur during the routine 8 hours operation hours. If possible, we will make a daily wrap-up meeting to discuss any concern or issues that were identified during that inspection day. The closing meeting is conducted on the last day of the inspection, and we will discuss the written 483 observation items and additional discussion items, and the firm will have the opportunity to make your comment, objections, and then we will issue FDA 483 observation form, and every inspector will sign, and we will obtain a copy of signed 483 observation form. Then we will leave. So major system covered during CBER inspections include 5 or 6 systems out of the 7 major systems listed here. Quality, facility and equipment, production, materials, packaging and the labeling, laboratory control systems. The last one, donor eligibility, only apply for some human cell and tissue products, and are often covered as a review item instead of inspection items. For each major system we inspected, we will focus on 3 critical elements. First one is written procedures, and second is your training program, and the third one will be records. Quality system is one of the most important systems covered during CBER inspection, as always covered in FDA compliance inspections. Quality provide oversight to all other systems and assurance of overall CGMP compliance for the entire manufacturing site. The quality may have a QA and a QC, two different units. We will look at the approved written procedures and documentations, including but are not limited to site master file or quality manual, quality agreements, training and the qualification program, procedure and the protocols, and the deviation management and the CAPA management protocol, change control, document control procedures, especially batch records, and the labeling control. Facility and the equipment system are important system covered by DMPQ inspection. During inspection, we will look at your facility design in terms of prevent contamination and cross-contamination to see if your facility provides adequate space for orderly manufacturing activities, provide adequate segregations for different manufacturing steps or different product manufacturing. We will look at the flows, why unidirectional flows are preferred, but if it's unavailable, we will look at your temporal segregation and the procedure control to see if those are adequate to prevent contamination and the cross-contamination. Your facility should provide a controlled environment, and your clean room interior surface material should be suitable for cleaning and should be smooth, impervious, unbroken, and cleanable, resistant to deteriorations due to repeated cleaning. All joints should be sealed, and you should have a coved corner for easy cleaning. We will look at your HVAC system, and this we'll talk in the following slides. We will look at your major equipment qualifications, including process performance qualification, and your equipment preventative maintenance and the calibration. We'll look at your PM protocol schedule and the records. We will review your facility and equipment cleaning in terms of your cleaning agents efficacy study, cleaning procedures, routine cleaning schedule and the records, and your routine cleaning monitoring. The HVAC system using the HEPA or upper filtration to provide a clean air to your clean room areas. Considerations of appropriate clean room classifications, including based on multiple factors, including but are not limited to whether you are a single or multi-product facility. And if you're multi-product facility, you perform concurrent manufacturing or your camping-based manufacturing. For all the product contact equipment, we will look at if it's a closed system versus open operations. For air handling units, we'll look at whether you use a dedicated or shared air handling units for different clean rooms. It depends on whether you have a single pass air or recirculated air. Airflow and the pressure differentials should be monitored with a warning system, and they should be recorded. We look at your HEPA filter certification, recertification records, and we will look at your HEPA filter repair and the replacement policy and SOP. Utilities, we will look at your qualification of water systems and the utility gases, and your routine monitoring of your utilities, including chemical and the microbial monitoring. Your building monitoring system and should be we'll look at your BMS recordings, and we'll look at the alarms, notification, and the calibration procedures, SOPs, and the records. For additional computer system used for data management, deviation management, the CAPA, and the material inventory, test and sample tracing, we will look at the validation of those computer systems. Environmental monitoring. One thing should be noted that EM, routine operational EM, should be differentiated from EMPQ, because after the initial EMPQ, clean room requalification is performed after shutdown or a major modification to clean room areas. However, routine operational EM, including viable and non-viable air particulates and the surface sampling, should be performed in clean room areas based on applicable guidance. We will look at your EM SOPs, EM sampling plan, and your EM sampling plan should be supported by a risk assessment. It should be noted that the EM sampling should be always performed by qualified EM personnel. We will look at your acceptance criteria based on recommendations in ISO 14644, Annex 1, USP 1116, and the FDA guidance. For your EM records and training, during inspection, we may request routine EM data for clean room temperature for a certain clean room, and temperature, humidity, and the differential pressure differentials. We will look at your EM air sampling, non-viable air particulars, viable air, and we will look at the EM surface sampling for viables, facility surface, equipment surface, and personnel monitoring. We will look at the EM deviation, investigation, and the CAPA associated with deviation. The evaluation of your contamination and containment control based on the review of all the followings, including facility design, procedure controls, and facility and equipment cleaning, and routine EM and the pest control. For production system, We will observe critical manufacturing processing steps. We will review your manufacturing process validation record, including batch record for conformance lots, sampling and testing SOP in the records, small equipment calibration status, and the small equipment the pre and the post-use cleaning records, and any deviation associated with manufacturing process. The MPQ will review the aseptic processing studies. We will request the operator gunning qualification personnel monitoring records on site, and we will review your most recent smoke study for grade 8 areas, whether it's a BSC or an isolator. We will review your most recent aseptic processing simulation study report. We will review your reprocessing rework written procedures. For manufacturing material systems, we will review your warehouse inventory system, storage, distribution SOPs, and we will look at if your warehouse is adequately segregated, have a segregated, separate areas for quarantine and rejected materials. We will look at your inventory record. Product office, product specialists will look at your incoming material testing and releasing, and you should have a risk assessment to support your incoming material testing program. And please remember that identity tests for incoming materials are required by law. We're looking at your shipping validation and shipping documentation. For gene and cell therapy products, chain of identity and chain of custody are very important. For the laboratory control system, your QC lab should be separated from your manufacturing areas, and it has physical separation with a separate entrance. While the QC lab is often unclassified environment, it should be clean and controlled environment. And you should have a grade A, BSC, or an isolate for any open and aseptic operations. We will review your SOP and the records for QC lab equipment qualification and calibration, and your analytic method validation and acceptance criteria. The testing of in-process drug product and EM samples. We will look at any OOS and investigation. Labeling and packaging. Sometimes you have a different facility to perform labeling and packaging. Then we will review your approved written procedure and documentation, especially about the label storage, printing usage, and the reconciliations. Your line clearance and controls to prevent the label mix-ups. We will look at your labeling, packaging, equipment qualification, and process validation. For gene and cell therapy products, the labeling and packaging often are performed under frozen condition. So we will look at your temperature control during the labeling and packaging process and your label adherence study. Now we will discuss some of the most common observations. In general, There are more observations for quality, facility, and equipment, and production systems. This is not just unique for. This is a common to all biological products. Please note that observations were identified by individual inspectors. They are not represent a final determination of compliance of your facility. And you can discuss any observations with inspectors during the inspection and in your response to the FDA Form 483. Common inspection issues including procedures. There were no written procedure for certain operation, or written procedure for an operation incomplete or deficient. Written procedure for an operation are not followed. And the documentations, including good documentation practice, are not followed. Data are not adequately secured, maintained, and managed. So here are the examples for quality systems, observation examples. Deviation management. Deviation were not initiated. And where you have several same type of deviations, you don't have a deviation report. Lack of complete root cause analysis. Investigation of OS of one batch was not extended to other batches of the same product. And timeline for closure of non-conformance is not justified, especially when it passed the required time for closing the deviation. For CAPA not effective. Sometimes when a CAPA was implemented, but same type of deviation occurred again. Lack of time frames for CAPA implementation, lack of evaluation of CAPA effectiveness, and the closing of CAPA. So sometimes we see a CAPA was open for a very long time. Training, aseptic technique training is inadequate, and this happens when you have a training program and the qualification program, and your qualified aseptic operator still have issues with aseptic process operations. Document control, data not adequately secured, maintained, and managed. And this often happens when you have a hybrid system. You have both paper records and computer-based records. So this can be issued. Who has access to the data and do the modification? Permits to do the modification of the data. Quality oversight, lack of a quality oversight. Sometimes you happen and you say this operation is responsible it's conducted and by different department, and in the quality review and the sign off of the records was not there. Change control. Change of certain equipment without the change control. This often happen when you change equipment after you make your PPQ lots. Facility and equipment system observation examples. Most common observations are for EM and EMPQ. Lack of air monitoring, viable and non-viable of clean room areas, for example, grade A and grade C areas. lack of service viable sampling of grade C pass-throughs or grade A BLC post-operations. Sampling size did not represent the size where most critical operation took place. Cleaning. Cleaning procedure for equipment and facilities were not specific enough to follow for consistent executions. Equipment qualification, that sometimes qualification of certain equipment has not been performed or is deficient. Again, this happened when you change your equipment after your PPQ lots was made and you didn't have a requalification of or qualification of your new equipment. Calibration and maintenance. Unplanned maintenance was performed without the quality oversight. Disinfectant effectiveness study was not performed or is deficient. Sometimes you may change your disinfectant, but you didn't perform this disinfectant effectiveness study again for the new agents you're using. Production system observation examples. The batch records do not document the completion of each significant steps, or the batch records do not document that all critical in-process parameters were met. Sometimes you have in-process test and you may not have the result immediately, but your testing records should be attachment of your batch record and should be readily available for inspection during inspection. Aseptic process simulation. Do not simulate all the critical aseptic process execute during the production. Sometimes you do not including all steps, or you do not including all critical interventions. Gross promotion tests were not performed, and this is important. Without gross promotion test, and your aseptic simulation data is inconclusive. Process validation. Your PPQ study was based on clinical batches prior to defined commercial process, and changes made to the commercial manufacturing process were not evaluated during the PPQ study, and those two are associated to each other. So you make changes after your PPQ lots were made. You do not have a validation of your changes. Reprocessing rework. Sometimes we've seen the reprocessing was performed without the written reprocessing procedure. Material system observation examples. It's an income material testing. So failure to reject lots of components that do not meet the appropriate written specification. For example, your plasmids below the concentrations below the specification, but you still use it at risk. Supplies COA for components or consumables used during the production are not periodically verified through the testing of incoming lots. And again, please note that identity tests of incoming material are required by the regulation. Inventory control and storage. and your warehouse do not have a clear labeled segregate areas for incoming material or for quarantine material or for reject materials, and they all receiving areas are overflowing with receiving materials. Laboratory control system observations. Examples including the suitability of all testing method is not verified under actual condition of use. There were no document system in place to track and manage the flow of samples. For gene and cell therapies and COI and chain of identity of material in the sample, very important. In process and release specification, so we see that something called testing to compliance. Retest of bulk and the final products were performed before initiation of manufacturing investigation of the OS for cell counts and the viability. The sample was retested in triplicate with passing results, and the lots was released. Labeling packaging system observation examples. The final packaging inspection, visual inspection parameters has not been fully defined. VI reject rates is not based on suitable statistical procedures. For gene and cell therapy, when VI failed and the re-inspection of the temperature-sensitive product could not be performed, no alternative strategy or remediation plan was development. Shipping validation is inadequate in that the temperature during the shipment does not represent a worst-case temperature challenging. The other thing is shipping temperature and the container closure integrity are not verified properly upon receipt during shipping validation. Shipped plasmas were labeled incorrectly. Material failed identity test upon receipt. Lack of control of the labeled templates and the excess printed labels. Another example of observation is that excess printed labels returned from production are stored in an unlocked cabinet. In summary, we covered with the CBER inspection, and the CBER inspection was conducted by DMPQ around middle cycle review clock, and all observations must be resolved before the approval of your submission. We covered major system and important aspect of elements covered during inspect during POA and the PAI. We went through common observation identified during the inspection. So acknowledge, I would like to thank DMPQ management and DMPQ authors for previous inspection presentations. With that, I would thank you for your attention. I would take questions after.
Dr. Wang, thank you so much for your presentation. It was very interesting. So we will now start with the Q&A session. And the first question is, if a firm is inspected by FDA CBER, and this could be a pre-license inspection or a pre-approval inspection, can an Office of Regulatory Affairs surveillance inspection be waived? Especially, the question is asking if the two inspections will be conducted within a few months.
The last part of the question was cut off. I think the question is which inspection can be waived. CBER can waive a CBER inspection based on a previous ORA inspection. But however, because the difference between CBER inspection and ORA inspection is CBER inspection is a product specific, whereas the ORA inspection is a GMP compliance of an entire manufacturing facility, including different products. So usually CBER ORA inspection is not likely waived after PLI or CBER inspection. Even if your product get approved, and doesn't mean that ORA will not find any compliance issues. Thank you.
Thank you so much, Dr. Wang. And the next question is, when does CBER announce the inspection if pre-approval inspection is needed for a BLA?
One is inspection is needed for BLA. We usually, like a presentation said, we do our CBER inspection around the mid-cycle review clock, and we will contact a firm or facility at least 6 weeks ahead of our inspection. So you will have enough time to prepare. Thank you.
Thank you for this response. The next question is, will the documentation and facilities of the production of historical batches, which were not manufactured in the proposed commercial facility, be inspected also during a pre-approval or a pre-license inspection? So the question is asking, if the products were not manufactured in that specific facility.
Well, CBER inspection, POI or the PI, is the facility that you have manufacturing your PPQ lots, and there's a facility where you manufacturing your commercial lots. So if you have a facility that manufacturing clinical, early clinical phases, clinical 1, phase 1, phase 2, we're not likely to inspect this facility, not in POI. Thank you.
Thank you so much for your response. We received a number of very interesting questions. And the next one is, if the drug substance and drug product manufactured at different sites, how FDA decides which site need a pre-approval inspection?
This is a good question. There was multiple factors we're considering. But first of all, your drug product facility, manufacturing facility will definitely be inspected. If there are no previous inspection history and it's a new product or it's a new facility. But drug substance, it depends. We will have to see what kind of drug substance and if it's already made almost just like a very little manufacturing to drug product, or it's still, it depends on the stage. So it's going to be case by case. Sometimes we will inspect both drug substance and drug products. That's happened before. Thank you.
All right, excellent. The next question asks, what is the requirement for visual inspection of cell and gene therapy products?
This is a question, very good question for product specialists to answer, and they may have a lot to tell you. But for me, based on our inspection experience, and then they all often inspect the drug product in the final container. Each company have their own specifications based on their drug products quality, and usually they have their history, and then they knew what should be look like a good product, and what see what kind of thing when they saw it during visual inspection is problematic. So it's a very depend on each product. Say, for example, if it's a clear, if it should have a opaque color, and if labeling was correct, if it's the seal of the drug product. A lot of time gene therapy products are filled into flexible containers, and you have a cracking on the container if there are any defect on the containers. So those are including in the usual in the visual inspections. I hope I answer your question. Thank you.
Thank you so much, Dr. Wang, for addressing this question from your perspective. And the next question is asking if the status and movement of materials, components, intermediates, and final drug products in a warehouse is controlled by a validated ERP system. Is it expected to also have a physical segregation of rejected, returned, or quarantine materials of components or final products?
Yeah, the very good physical segregation is always expected. Just because we'll take it in and put it in material are human beings, and the ERP is a software system. So in case anything goes wrong, if you have a good physical segregation, that will reduce the cross mix-ups. So the answer is yes.
Thank you so much. And thank you so much also clarifying the ERP system and what it is to our audience. And we are coming towards the end, and this will be our last question. For changes to the manufacturing process after PPQ, is the expectation that a full PPQ after change implementation is performed, or can a risk-based approach be applied to assess if requalification of the portion of the manufacturing process directly impacted by the change is sufficient?
Well, after change, a process validation run, it depends on 1 run or 3 run is always welcome or per se required. And this is, I think, a product office reviewer can chip in. So sometimes it depends. Sometimes if the change is a minor, and you might have a justification why you didn't perform a full run of process validation. But this is a case by case, so it depends on the change.
Yeah, thank you so much for addressing that. And we really appreciate your time today and answering audience questions. And I will also like to take the audience for attending this session and asking this engaging questions. And I would like to close today's session, and I will also will turn the microphone now to Dr. Lapteva for her concluding remarks for the conference. Thank you.
Thank you, Zehra and Wei. The topic of inspections is always of interest to our stakeholders and attendees, and we're glad you were able to participate with us today and talk about inspection-related issues. And now we are at the end of our second day of presentations from the Center for Biologics Evaluation and Research. And we are ready to conclude our entire week-long conference with participation of staff from Center for Evaluation and Research, Center for Devices and Radiological Health, and Center for Biologics Evaluation and Research. And I would like to thank all of our presenters and moderators for sharing their expertise and perspectives, as well as our planning committee and the SBIA program staff, and of course, Brenda Stoddard, who every year brings us together to organize this event and deliver a new educational program, which we hope will help you, our attendees, plan your product development and your future regulatory submissions. And of course, very special thanks should go to the conference support team for all of the preparation and for the excellent job that they've done during this event with running it in the virtual environment. On behalf of the Center for Biologics, I would like to express our sincere gratitude to our sister centers, CDER and CDRH, for their support and collaboration. And most importantly, and I say this every year because this is truly the most important gratitude statement, thanks to all of you who attend and participate in this conference. Thank you for your engagement, for your interest, for your questions, and for your evaluations and feedback. We hope the contents of this conference will continue to meet your needs for the regulatory educational experience. Now, if you missed any of the sessions or presentations in this week's event and would like to view again some of the sessions or maybe most of the sessions, you can access our FDA website and the event site, which you should see right now here on the slide. The post-event viewing is not subject to any time restrictions like CE credits. Therefore, you may view any of the sessions at your convenience. And of course, since the Regulatory Education for Industry Conference is an annual event, we hope to see all of you and other participants at our event next year. Now, when the pandemic is over from the FDA side, we hope to run the next event as an in-person or at least as a hybrid event as we carry on with our educational series on more topics important for new product development in the future. We invite you to join us again next year, and we look forward to seeing your evaluations and recommendations for new themes and new topics to include in our next year's conference. Once again, thanks to everyone for joining us this week. We wish you all a restful weekend and a great year ahead. Thank you.